Workshop, Recent Trends in CAR T Cell Research and Manufacturing. As most of you have noticed, we have a very interesting mix of speakers from academia, research labs, and industry, which will contribute to different flavors on the topic. My exposure to CAR T came from a very personal experience where my sister was diagnosed with leukemia and she was going through various options available for her treatment. So that is when I did a little digging to get some idea about it. A very common bad habit with us researchers. We try to research everything, whatever we lay our hands upon. Since I was not in the allied field. So even though a few therapies have been approved by FDA, immense research and trials are going on globally to effectively use CAR T in mankind's fight against cancer or blood cancer, to be specific at this time. Also, because CAR T targets specific antigens, solid tumors still pose a challenge due to their heterogeneous antigen milieu. And there is emerging research into how nanotechnology can be used to improve immunotherapies, including T cell therapy, due to the inherent capabilities of nanoparticles to deliver therapeutic cargo, enabling both imaging and therapy of the solid tumors. So I see there are a lot of work going on in that area. So we will definitely see a quantum jump in improved cancer therapies in the coming years due to the advancement of efficient immunotherapeutic techniques. I'm sure the participants will have an enriching experience and will go back with better clarity on the challenges as well as the possibilities of CAR T therapy.
So now I invite our first speaker, Dr. Albina Nizar from Tata Memorial Center, Mumbai. So Dr. Albina had completed her PhD in biochemistry from University of Kashmir and has been associated with Tata Memorial Center since 2018. So I invite Dr. Albina Nizar once again and provide us some insights on the recent trends in CAR T research and manufacturing.
Hello, everyone. I'm very happy to talk here. The topic of discussion for today is and give you an idea about how we are right now, what is really required for CAR T therapy, how we've reached where we have reached, and what is the next ahead, and how, if people want to get into CAR T therapy and cell therapy, this presentation can be a starting point for all of them. To sum it up in one slide, we pioneered the CAR T cell therapy in India with the first patient infused in first in human CAR T cell trial in June 2021. And we currently have been able to enroll around 16 patients, 10 from DLBCL and 6 from a BALL. It was a historic event in the entire cancer field in India. And as you can see, this was the first patient which was infused for BALL, and it was covered largely in the news. And the first patient was done for BALL, and shortly after, I think about a month later, Dr. Hasmukh, which you see here in the middle, did it for BALL. This is Dr. Gaurav and Dr. Rahul. You're pretty much familiar with the faces already. Two interesting things about our project. We have been able to keep cost marginally very, very low, brought it down from about 4.75 million for Kymriah and Yescarta to around 30,000, 31,000 US dollars. That is what we are aiming at. And we have been able to go from bench to bedside in a remarkable period of just under 5 years. This is where I really want to get into the explanation of what is CAR T cell about. So as you see, this is a picture which is actually taken from the NIH website, and you can see two lymphocytes basically attacking a CAR T cell. And the lymphocytes are actually the host cells, as we all know. And this is the power of immunotherapy, that we can actually take, it's an autologous therapy, and we can actually take the cells from the host's own immune response and use it to fight against the cancer. So, just a basic if overview of how CAR T cell therapy works. So, we call it living drug. CAR T cell therapy is a living drug because for the product development is actually taken from the patient and then given back to the patient itself. So the basic steps that are involved start from the leukapheresis, which is the sample is taken from the patient. Then the sample is ex vivo genetically modified. We have T cell activation and we transduce it with the required gene vector, the CAR construct of interest, which in our case is a CD19 CAR construct. Once transduction is successful, then we expand the cells with CD3, CD28 beads, which are already added before transduction, of course, with the growth factors. And then obviously product characterization and assessment of what is the quality of the product that you have formed, and then finally given back to the patient. Sample collection and the infusion, the patient also undergoes preconditioning chemotherapy and other different lymphodepletion strategies, obviously, which the clinician decides and which I'm not going into. So, this is the structure of a CAR construct, which we have a first generation CAR construct and a second generation CAR construct. Uh, this is I'm not going into the detailed structure of the CAR, but as you know, the difference between the first and the second generation basically lies in the co-stimulatory domains. So the first generation has one co-stimulatory domain, which is three. So here in this case, what I'm displaying is CD3 zeta, and in second generation, there are two co-stimulatory domains. We have CD28 or we have 41BB, and then you have CD3 zeta. And we also now have three generation generation CARs where we have three co-stimulatory domains. This obviously co-stimulation basically implies to more activation and killing efficacy of and identifying the structure and the binding with the desired tumor antigen. As you know, the CAR structure has an extracellular domain, the SCFP region, which recognizes the target antigen. There are intracellular domains, CD3 zeta, which is for intracellular signaling, and 41BB or CD28, which is basically for co-stimulation. Then we have a spacer and a transmembrane domain. This is the general basic structure of a CAR construct. This I've already gone through. First generation CARs with only single domain, second generation CARs with CD28 or 41BB, and third generation with CD28 and 41BB. So this is again a platform for a diagram basically giving the various different steps in the CAR T cell manufacturing involved, much more detailed manufacturing steps. So we start from the product fractionation. There are different strategies which you use when you take a product and then select differentially the different cells that you of interest, your cells of interest into that product. We have different technologies, as you're already aware. Probably most of you have used them also. We have immunomagnetic beads, which help us to select the cells, and also now there are immunomagnetic beads, which can be used in combination with different instruments. We have Dynabeads from Thermo, and we have Miltenyi beads. We select the cells, and then we proceed for activation. So a lot of combination of beads and consumables, which is very technical, and I'm not going into details here. What I can say is that there are many consumables broadly based on immunomagnetic bead selection and equipments which are cell therapy grade, like from Miltenyi, Clinimax, which all help you to select the type of cells that you are interested in. In this case, the T cells, CD3, CD28 are the most commonly used beads. Post T cell selection and activation, then of course you go for the gene transfer. There are different types of of vectors which are used. There are two products currently which are more than two products. I'm talking about 2018 when the first two products got approved. One was retroviral based and another was lentiviral based. So both of these are very common, and some of the companies use retroviral, some of the companies use lentiviral. Ours is a lentiviral based product. And through a lot of different systems, through traditional system of spinoculation, through transposon systems, through electroporation, gene transfer is done. Once transfection efficiency is we determine through different assays, we proceed for expansion. Of course, we do not wait for the assays. It's just a characterization step there. Once we know we have optimized our process development, we usually do not stop at the transfection step and we just keep on moving with the expansion towards the expansion. There are different platforms which are used for expansion. Post expansion, again, I'm not going to get into the instrumentation part of it because that's beyond the scope. Post expansion, we go into washing and change of the media and basically designing the D cell formulation. A lot of different, again, instruments are used there. Then finally, when we have the desired dose which is required, we go either for a cryopreservation or a fresh infusion. And then the patient is for cryopreservation, we use different types of cryopreservation reagents and techniques. Most of the common and the most desired one is control rate freezing. It helps to maintain the cell viability post thaw, and finally getting the product back into the patient. This is the Prodigy instrument, which is it's like a one-stop solution. This is a completely closed automated system which is able to manufacture CAR T cells within a period of 14 days. It's a complete closed circuit system, which means there is no, these are all tubings, and the product actually, the product that you see here actually moves through different tubings, column-based process. It is able to select the cells, transfect the cells, then expand the cells, and finally have the final product for infusion, which you wish to cryopreserve or freshly infuse. It's your choice. The challenge is when you begin with the CAR T cell therapy and the basic benchmarks or the checklist which should be there in CAR T cell therapy is that you have to practice and you have to produce CAR T cell manufacturing as per GMP guidelines during the whole process. There are basic facility considerations apart from the process development for the GMP, which includes your facility design. The processing should be aseptic. There is environmental monitoring, control for cost contamination, qualification and validation of facilities, instruments, and process. And of course, there is regulatory overview. What I really want to highlight here, and the reason the slide is here, is basically to make you understand that during this whole process of manufacturing of CAR T cells, what is really crucial and what defines what the product will go into the patient or not is product characterization, product release testing, which is based on different assays that we do to ensure that the product is basically within the limits of acceptable criteria for release into the patient, which includes viability, cell count, phenotype, and sterility. So the regulatory considerations, when we started in 2018, there were no GMP guidelines. There were no, not GMP, there were no cell therapy guidelines in India, and we were following US FDA and EMA guidelines to get the regulatory approval. So we set our benchmark really high and we followed all US FDA and EMA guidelines. And in 2019, the first cell therapy guidelines came out, of which we were a huge part. And we submitted our comments and suggestions to them. And now the 2019 GTP guidelines are in force, and that's the guidelines for cell therapy currently in India. The facilities which are required for CAR T cell manufacturing are very specific. We need sterile clean rooms. As you see, this is a clean room. The type of the product and the makeup of the product would decide the type of GMP that you want to have. For example, in case of, if you, for example, hypothetically, we don't want to make CAR, we want to work on COVID, then obviously we need a higher benchmark, as we all know, for the sterility, even in clean rooms. So an ISO 6, a negative pressure room, would be applicable for that. For CAR-T, an ISO 7, positive pressure room is what is required. The stage and the scale of the manufacturing, whether you're going for local production, whether you're going for commercial production, would actually define the type of the clean room. The personnel and the material flow, what kind of product that you have, universal direction of the clean room design. It all depends on the type of the product you want to go for, allogenic or autologous. Then this is something related to the AHU handling, whether the AHU is concurrent or campaigning, and of course the cleaning and the personal SOPs. Finally, this is a glimpse of just to show you what we were in 2018 and 19 when we started the manufacturing the facility. And this is the team now, the first CAR T cell center in the country in 2019. This might give you an idea of what our facility looks like. And this is Dr. Gaurav, who's the head of the CAR T cell center. And we have postdocs and JRFs, manufacturing associates, quality control associates. So some of the team you can see here. Acknowledgements to all the cell therapy based companies who have been helpful for designing our process development approaches, and for NIH support for being our intellectual partners. I'm happy to take any questions from here on. Thank you.
Is there something else in the market on electrochemical based, like how you call Max? Is there any other cell sorter
There are many
you are
cell sorters. There are
that
now
you're
in
planning to
2000.
purchase or you're using it?
No, we already have many different platforms which for manufacturing, and most of those platforms are all customized and designed by our own in-house team,
Okay.
and many of them are under on the way to patent also.
Okay.
So when you say platforms available in the market, there are a lot of different platforms which are available for different steps. So we have platforms for cell selection, we have platforms for transduction alone, we have platforms like Prodigy now, which
Yeah.
can do the whole thing completely. So we have competitors, so to say, for Prodigy also, and we have semi-automated platforms for which I'm a big fan, which can do steps of cell selection separately, followed by cell washing separately, followed by expansion separately. So there are different platforms available in the country and globally also. But I think most of the people, the other centers in the country, are following the Prodigy way, and they're doing the manufacturing on Prodigy. We at our center, because we started very early on, we were interested only to keep the cost down, and in that sense, we wanted to keep the process development and the manufacturing indigenously designed, and we have different manufacturing platforms where we have indigenously placed the whole platform in a way to keep the cost low. We have all the infrastructure and the equipments available. Many of the equipments that I told you are already in our facility. So we have already these equipments, and we have different manufacturing platforms.
What are the recent trends and emerging trends in biopharma today? Now, I do not want it to be, something that I just keep talking and you don't get value out of it. Let's have it more as a storytelling, because without context, no content can be valuable. So I'm here to give you a little bit of context as to where do these cell and gene therapies in general and CAR T therapies in particular came into existence. And hopefully, this can set the tone for you all to understand that when we say CAR Ts and when we say curative therapies, it is a little more beyond than the conventional pharma. It needs a lot of dedication. I'm not saying that other fields do not require dedication, but as we go through the story, probably you'll understand that what do I mean by perseverance, dedication, and tragedy, striking multiple patients, and something that brought CAR T striking multiple patients, something that brought into the fore and into the element that we know today as therapy. So what I basically mentioned in the tagline of this particular presentation was, what are the emerging trends in biopharma, and what has led to the rise of cell and gene therapy in general and CAR T therapies in particular? So when we look at the ecosystem of biopharma and look at what the disruptions are, there are primarily 5 factors of disruption. And I mean, these disruption factors are different for different industries. So when we talk of traditional biopharma, 5 things that can disrupt traditional biopharma are: number one, prevention and early detection. And that is where you see a lot of scientific movement also happening in that space. There is a lot of impetus on biomarker identification these days. Neonatal screening, so that kids, when they're born, can be screened for the diseases, and those can be addressed very early on in their lifetime. The second primary cause of disruption is digital therapeutics. So primarily in the space of, I would say, psychosocial kind of therapy, this is creating a very big disruption in the biopharma space, where a patient can get access to therapy not by visiting a doctor. So the patient does not have to go through the social stigma that, oh my God, I feel that somebody would recognize me there, and I'm not able to approach the right kind of intervention. But with digital therapeutics, that gap is kind of narrowing now. And what also we are seeing in digital therapeutics is that the track and trace is also improving. Then the next thing is around customized treatments. So when I say customized treatments, it is not one size fit all. We all are humans, and being scientists and being in the field of biotech, you all would realize that even though most of us share 99% of our 99.9% similar kind of a genetic makeup, there are still nuances that make each of us different. Be it our sex chromosomes, XX and XY, be it racial differences in a little bit, but all of us respond to treatments and certain interventions differently. Something that works for me may not work for you. And that is something that science took a very long time to understand. And the third thing, I would say it's fourth thing now, is curative therapy. Till now, our major inclination in terms of biopharma has been in treatment management. I would say symptomatic management. So, okay, so if you have diabetes, oh, your blood sugar rises. Why not take something that is kind of repressing your glucose markers and bring that blood sugar down? And even if it's down, you just mask it. Similarly, for hypertension, we kind of induce the vessels to dilate a little bit so that the blood flow is kind of regulated. But we do not kind of treat the root cause of it. And therefore, this idea of curative therapies kind of opened a Pandora's box, I would say. It was a key to a new kind of a distinction. And, nothing happens in a day. When we say, and I would cite a very recent examples of COVID therapies. So when Pfizer and Moderna came up with the mRNA therapies, it was not as if all of that happened in a day. They basically build up their mRNA sequences. Even the lipid nanoparticle vesicle in which that mRNA was kind of delivered into the body was built on almost decades of basic research that was there. There were hundreds and hundreds of papers that outlined what is the usage of these. And lipid nanoparticles, because their toxicity and safety were established in other therapies, could be leveraged here. Imagine a situation where none of that would have happened. We still may have been in the lockdown age. And now we are progressing towards, let's say, the end of COVID-19. So with this idea of curative therapies, let's move on to the next thing. Let me take you back to a story from where CAR-T therapies actually started. So let's begin. Let's imagine that there is a 5-year-old child. So this is the story of the first pediatric patient, or what we may like to call it, patient number 1 for CAR-T therapies. Her name is Emily Whitehead. What you see on the left of your screen is Emily Whitehead when she was diagnosed with ALL. Now, ALL is a hematopoietic malignancy, which is like a heme malignancy, blood-related cancer. So in 2010, within a span of one night, Emily Whitehead transformed from being a perfectly healthy child to a cancer patient. So it was in 2010 that her parents kind of got informed that Emily has, ALL. And the pediatric patient said, quote unquote, if you had to have a kid with cancer, ALL was the cancer to have. Because until then, it was assumed that, ALL has a fairly good chance of prognosis, so it can be kind of treated, especially with bone marrow transplants. So the doctors put her on the usual regimen. There were 2 rounds of chemotherapy, but then nothing worked. The cancer still persisted. Then a bone marrow transplant was recommended. But just like everything else, even bone marrow transplant sometimes works for patients and sometimes does not work for patient. And the reason is that your own cells start triggering your immune system to an extent that it can become lethal for your own self. So the bone marrow transplant was not taken up as a consideration by her parents. She instead was taken to a facility called as CHOP, Children's Hospital of Philadelphia, I think it's Pennsylvania. Yeah, so it's CHOP, where her father learned that there's this scientist who has been working on a very experimental therapy, which is trying to manipulate the body's own T cells and then allowing them to fight cancer. And because his father, I mean, any father would do, even our fathers would do for us, he was like, I want to take my child to CHOP and see what they can do. But everything's not as straightforward. FDA by then had not legalized the therapy or had not approved initiation of trial. So what that means was Emily Whitehead could not receive that therapy. However, as time progressed, we are already 2 years out from that, and Emily's condition is deteriorating. Her doctors have given her a 3rd round of chemotherapy, and that has failed itself. So after that, the doctors were like, we don't think we should kind of put the child through any more pain. Why don't we recommend hospice to her? And let me tell you what hospice is. Hospice is we stop all the ICU treatments, and it is only going to be pain management for the patient, which means no more treatment. And you just wait for the patient to die. And imagine this happening to a lot of patients over the course of years. But Emily got lucky. In 2012, when her father's incessant kind of reached again back to CHOP, and Dr. Carl June, the eccentric scientist I was talking about, who was still working on CAR T therapies, got the approval, got funding to start the CAR T therapy trial. That's when she was the first pediatric patient who was infused with this CAR T therapy. Her blood was drawn out, and I'm sure you must have heard of the process several times, and you will keep hearing about the process, I think, over the 2 days. So I'll not bore you with the context. So her cells were taken out, processed, the T cells primarily were genetically modified and then reinfused back into her. It sounds pretty easy, right? But it is not. What happened after that was more scary for the scientists, as well as for Emily's parents. She started showcasing a very high inflammatory reaction. So she ran very high fever, she was not responding. Basically, she went into a coma. Now, what that means for any scientific study, let me give you a little bit of background. If any patient dies in a clinical trial, it is referred to as a grade 5 toxicity, and which means that the trial has to stop. And no more patients would be ever infused with that therapy. So had Emily died, please assume that what you and I are talking about CAR T therapies today would not have existed. It would have pushed the field back by at least a couple of decades. And this is very evident from what happened to gene therapy. Way back in 1990s, I think Dr. Wilkins' lab, again, from CHOP, Pennsylvania, he had started doing some gene therapy experiments. But 2 patients died in that trial, which meant that FDA stopped all sorts of funding for gene therapy, and gene therapy kind of was pushed back by at least a couple decades. So Emily was also on the deathbed, remember. Nothing was working. And doctors then did not know, because T cells are not supposed to trigger other cytokines. So what they did was they sent her blood to a lab. Now, usually labs in those days, we are talking a decade ago, takes around 2 weeks to come back with the result. And the doctors literally had to beg the diagnostic lab to send the results early. When the results came back, right, so they found that there is a particular cytokine that's overexpressed exponentially. And what I mean by overexpressed exponentially is the IL-6 was almost 1,000 times overexpressed in her blood profile. And that made the doctors a little bit confused because T cells then were not supposed to induce interleukin 6. And so what do they do about it? And here I think serendipity works. So Carl June's daughter, was diagnosed with rheumatoid arthritis. And rheumatoid arthritis kind of is triggered primarily by an inflamed interleukin 6. So she was administered tocilizumab, which is, again, an antibody that targets IL-6. So Emily Whitehead, based on Dr. Carl June's recommendation, was given tocilizumab. And it was a miracle of sorts that she started improving right within 6-7 hours. And she came out of coma and has been almost 10 years cancer-free today. So that's her on the right, I think last year itself. And all her blood profile markers indicate that she's been doing well. So this is the story of, I would say, the tragedy, perseverance, and hope. Tragedy on account of a child being, diagnosed with a terrible disease. Tragedy on account of Dr. Carl June, who had seen his wife pass away due to ovarian cancer a few years back. That's when he started working on it. Perseverance on part of both Emily's parents as well as Dr. Carl June, who, in his own words, may have had to stop the research, several times, but he persevered. And hope, also again, on part of all the concerned parties, because hope is what drives us. So I hope this story resonates with you, because CAR-Ts and gene therapies and all the curative therapies that we talk about today are a little more than just something happening in the backdrop of your lab. They basically change patient lives. And in Emily's own words, so what you see on the left of your screen is a quote from Lisa Rosenbaum. Now, she's a scientist. So she says that the emergence of CAR-T therapies, like most scientific advances, reflects incremental insight of hundreds of scientists over decades, something that I was kind of alluding to early on in my conversation. You do not do it all alone. You can never do it all alone. When there are secret projects in the lab cannot move forward unless everybody is open and everybody is collaborating. I was a PhD student, so I always understand what it means. I would say that part of my PhD, I attribute to all my friends, my lab mates, and my mentors, because you, more often than not, run into bottlenecks. You don't know what to do next. And when you go out on your chai kind of break or your coffee break or your breakfast break, that's when you start talking to your friends and ideas come across. And that is what takes science forward. Now, moving on, what you see on the right is Emily's quote. So she says, her hope is that CAR-T therapy will become a frontline treatment option and can be readily available, because she's grateful that, she got the treatment and she got another chance at life. And as I read somewhere, I think a couple of days ago, that basically nobody wants people to watch them die. And these CAR-T therapies is the last ray of hope for some of these people. And that is why this is a disruption in the biopharma space. And so before going on to the next slide, I just want to put things in context, what we kind of spoke of till now, and what do we understand about it. So I told you, it was not that there was dearth of treatments. Emily Whitehead had undergone 3 rounds of chemotherapy. It was not that there was no pharma solution available. She was offered a bone marrow transplant, but nothing was working for her. So why CAR-Ts? Because it is a hope for people when nothing works for them. If, again, it's not like, if today, let's say, God forbid, I'm diagnosed with cancer and nothing works for me, I can go and seek a CAR-T therapy, because I have to be eligible for that CAR-T therapy based on some of my biomarkers and some of the treatment paradigms. So for some people who are eligible, this is like the last shot at life. So cells proliferate, they escape all the immune attacks, and that's how they cause what they cause. So there is a very high incidence of cancer, and a National Cancer Institute kind of pegs it as around 18.1 million new cases of cancers across indications. So here it is heme cancers, solid tumors, everything under the sun, all type of cancers. Around 18.1 million new cases are diagnosed every year. Now, the second reason is that support of regulatory and market access environment. Now, these therapies cannot come into market if FDA or in case of India, DCGI do not recommend them. They do not open doors and windows for these therapies to come out. So FDA has allowed some of these therapies, specifically in very late line cancer or gene therapies that target rare disease, to be accorded a special status called as accelerated pathway. So what that means is, if let's say traditional therapies take 10 years or 15 years in clinical trial before they are, brought on the table for discussion of whether to launch or not launch, these therapies, they have a very squeezed timeline. So these therapies, right from initiating the trial to getting into the market, may take as less as 5 years. So right from enrolling your first patient to then enrolling it in a clinic may take just 5 years. So that time frame has squeezed. And secondly, market access. So when I say market access, what do I mean? When I say market access, I mean that these therapies are covered by certain insurance programs. Now, folks can always pay out of pocket, and 60 to 70% of treatment in India is out of pocket. But for very expensive therapies like this, that cannot happen. So I'm not even sure if anybody can afford a 500,000 therapy on their own, unless they fall in the top 1% of strata. So in that case, your insurances come very handy. So some insurances, and in US, I'd say 70 to 80% of their population is covered by some insurance or the other, be it the federal insurance, which is Medicare and Medicaid, or their personal insurances. So these big personal insurance, private insurances, they also assess therapies and take them in their formulary. So that is market access, which allows patients to access these therapies. And these payers will eventually pay for the therapy. So this kind of support, when people saw that these therapies are giving special status by FDA, they are actually being included in formulary by these insurance companies, kind of provides an environment of optimism. And thirdly, but not lastly, growing awareness. So we today are talking about CAR T therapies. Imagine patient who becomes, an expert in his or her own disease. They may be searching several papers. They may be reaching out to several people. There are patient support groups that exist. So there are discussions that happen. The patient today is much more aware of their own disease than what somebody may be, let's say, 10 or 20 years ago. If you are diagnosed with any minor chronic disease, you kind of break the internet. You go in and you try to read as much as you can around that particular disease. So the generation today is more aware, and that also leads to more understanding of newer therapies. People are not hesitant to try a new experimental therapy. So these are the three key drivers. And again, people now also know how to manufacture these therapies. And that has happened because people were manufacturing biologics before this. And biologics is monoclonal antibodies. Some of the same systems have been translated to these therapies, and they are manufactured in a similar way. So how to manufacture viruses at a very big scale can probably be attributed to our vaccines. How to manufacture cells at very large scale can be attributed to, let's say, our monoclonal antibodies. But again, I would say that no instrument or no infrastructure exists today that is just for cell and gene therapies. We are trying to put together things.
Can you repeat? Can you repeat? There is no infrastructure?
So I would say that there is no dedicated infrastructure that is just there for cell and gene therapies. There are companies that are working in this space so that things get automated. Even today, 70% of CAR T manufacturing requires some kind of manual intervention. Right. It is not like you put your clothes in a washing machine and you forget it. And by the time you come back, it's ready. You would need cells, you would need to seed them, grow them, tweak them, monitor them, and then again populate them, check for all the quality assessments, do all the diagnostic tests, and then eventually thaw them, package them, label them, send them to the treatment center where it will be infused. So there are a lot of steps. And the current paradigm of manufacturing, yes, it's evolving. The first therapy was approved in 2017. It was Kymriah, the same therapy that Emily Whitehead received. Novartis acquired the rights of it, and then it was commercialized as Kymriah. So since Kymriah was launched in 2017 to now, there has been extreme, I would say, innovation in this space. But what the field lacks is still, some kind of technological platform innovation that will make the manufacturing easier, and that will also bring the costs down so that the patients can derive the most benefit.
Can you repeat, ma'am?
Yeah, sure. So I'm saying that, if the
Lack
of platforms, I would say, lack of dedicated manufacturing platforms, and the time it takes to manufacture these therapies is driving a lot of the cost. So if there are dedicated automated platforms with low failure rates, then it can bring the cost down, something that is scalable. What I mean to say is, when we do experiments in the lab, I can use the fanciest of sigma enzyme. I can use the fanciest of molecular reagent. But if I'm thinking of making the same thing for 2,000 people, would I still use a 2 lakh rupees enzyme? I need to find alternatives. And so currently, CAR T therapies are still not as scalable. By scalable, I mean easy to translate from bench to a 200-liter bioreactor. It's still not there. There is still a very high gap between what we do in lab to what can be taken out with the industry.
So is it because the scalability issues because of lack of manufacturing facility itself? Because I don't think we are lacking with any technology on manufacturing facility. Could be like, you develop the automated machine and you get it approved as a patent. The patenting process is quite long in India. Is that a challenge, or what could be the challenge? Because we are all very good at manufacturing things, if I'm not wrong, in India.
Understood.
We
Yeah, no,
have
that's a
very
great
good
question.
marketplace also, yeah. So
The
what should
problem is, Dr. Jagradha, that the instruments, or let's say the bioreactors we use today, are bioreactors that would traditionally be used for generating monoclonal antibodies. These are not bioreactors. So I would say most of the equipment that is used for CAR T therapy is a retrofitted equipment, which means that they were initially when they were created for something very different. And now scientists are using those equipments to create this. And because of, yeah, that's a great point. Another thing, because of these therapies, once a process is set and it is submitted to a regulatory agency or FDA for approval, it means that the process is set. You cannot change it. So let's say if I, as a scientist, I'm working in my lab and I created a CAR T therapy based on, your Sigma centrifuge, ultracentrifuge, your own bioreactor chamber, using, GIBCO media and all of that. If that protocol is set, the therapy will require the same protocol, even if it is required to manufacture for 2,000 patients, which means that the lab scale equipment is not equipped to manufacture it for 2,000 patients. And because when I submitted the protocol, I use those equipments, I will have to use the same equipments when I start using it for 2,000 patients. Another challenge in CAR T therapy is failure rate. So imagine a circumstance where the patient is dying. I somehow managed to take the right amount of blood from the patient, but then I found that the T cells that I isolate from the blood are not enough to manufacture, which means the patient is already very, very fragile at this stage. I'm not getting the right amount of cells. And what if I ask the patients to, undergo apheresis or blood collection again? The patient very well die during the process. So that is another problem. And like I told you, there is a little nuance around failure rates as well. So clinical failure rates and commercial failure rates are very different. So commercially, we see that there are around 12 to 15% failure rate, which means 15 out of 100 runs would fail. What that means is those 15 patients will not receive the therapy in time and can potentially die waiting for the therapy. So currently there is a very long vein to vein time. And this is all again due to multiple reasons, infrastructure, platforms, and squeezing the, I would say, doubling time. Novartis has recently come up with a platform called as T-Charge platform, which kind of claims that they can reduce the manufacturing time from 14 to 20 days from to just 2 days. So with platforms like these coming into the market, probably in future we'll be a little more capable of ensuring that every patient who is eligible to receive the therapy actually gets the therapy.
Background about Miss Beena ma'am. She is currently working as the CFO of Colorado Histoprep at Fort Collins, as well as the Chief Financial Officer of Care Research LLC, based in Fort Collins, Colorado, USA. Miss Bawa ma'am has earned her MBA in Finance and International Business from Rutgers University, New Jersey, and Program of Instruction for Lawyers from Harvard Law School. She joined the Xerox Corporation, Rochester, in 1984 as a senior financial analyst, and soon she was promoted to the role of controller, responsible for finance and accounting operations. Ms. Bawa ma'am founded Bawa Financials in the year 1996 and worked on providing commercial lending and financial services to emerging small businesses. Over to you, sir.
Very much. So first, I must apologize that instead of my lovely wife, you have to see my ugly face. That having been said, I don't think I can ever be a substitute for her. She's my better half. So I'm going to share my screen and then let's take it from there. Our talk today is financial challenges and considerations facing a small business in a competitive marketplace. So one of the things I wanted to do was share with you almost like a case history of what we had to go through to get from the conception stage into the marketplace and start earning money and providing valuable resource to the life sciences business. So this is a brief history of us. And the reason I'm going through this is because I wanted to share with you that most of the Indians in the US, they have impeccable credentials. And not that I want to boast or anything, but that I think if you want to get into the technical area, apart from selling newspapers or something like that, you need to have a good, solid technical as well as financial background. So briefly, after I graduated from BITS, I went to MIT. I did my 2 master's degrees. For some reason, they instead of calling it MS, SM is what MIT likes to call it to distinguish themselves. I did my thesis in polymeric drug delivery with Professor Robert Langer. And simultaneously, I did another degree as well, which means I did twice the amount of the course load, but wrote a common thesis straddling and satisfying the requirements of both departments. Subsequently, I moved to Hoffman La Roche, where I invented some innovative drug delivery systems. And then, since it was such a conservative area, and Roche was at the height of its peak with Valium, they were high on Valium, with a huge amount of money coming in, Valium being the largest selling drug in the market at that point of time, that they were very conservative. And so any new ideas that I would have, they would shelve. So I swore that I would never get into the pharmaceutical business again. And I joined Bausch & Lomb. Once I joined Bausch & Lomb, I was working in the contact lens area, and I realized that there's a wide open field as far as making shape devices for ophthalmic applications is concerned, because I could make shape bodies. The processing that Venkat was talking about, I did the process development as well as the product development. And then, of course, since my love was in drug delivery systems, I figured, okay, I can start putting drugs into these tailor-made polymers and shape bodies. Now, further downstream, I also developed a supercritical fluid extraction method, which for the processing of lenses, which saved the company millions of dollars. Simultaneously, I did my PhD at the University of Rochester, also in drug delivery. I was a fool. I could have stayed with MIT, and Professor Robert Langer told me that if I spend 2 more years there, I will get a PhD. But like an idiot, I wanted to go and see if somebody would pay me for all the stuff that I had learned. And in being an Indian, we have been in academia all our life. We don't get exposed to the real world. So I wanted to make sure that somebody would pay me for what I'd learned. So rather than following his advice and taking 2 more years and getting a PhD from MIT, I did it the hard way. I was working full time at Bausch and Lomb. I got my PhD there. Joined Alcon Laboratories in the ophthalmic pharmaceutical division of Nestle. And at one point of time, the shares of this were privately held again. And I was there from 92 to 93, provided them a platform technology for ocular drug delivery systems, which basically resulted in multi-million dollars. And we bypassed Merck's patent for timolol, which is a beta blocker for treating glaucoma. And I bypassed that, and again, that resulted in a pretty good revenue stream. Then I joined Atrix Laboratories, developed processes for anti-cancer agents, leuprolide acetate, for the treatment of metastatic prostate cancer. And these processes allowed for accelerated approval for formulations lasting about a year after a single dose versus getting an injection every single day. So by and large, I have about over 25 patents and several publications. And basically, I got tired of company politics. So I decided I'm too stubborn a person to work for somebody else. And I wanted to strike off on my own and see what I could do without the constraints of being working in a company. Now, this is a brief history of Veena, and of course, Sudhir has done a great job at this. Let me just fill in a little bit. Veena got her LLB called to the bar in Delhi. She was a legislative and election committee officer in the Indian Parliament in New Delhi. And what that means is when all these politicians pass these bills, somebody has to relate that and put it into law. And that's what this was doing. That's what she was doing. In addition to that, she was also election committee officer. And so even the members of parliament, including Indira Gandhi, when she used to come, used to get a ballot from my wife. And since she was one of the few ladies there at that position, he also got a very good smile from her occasionally. So then she got married to me and gave up parliament. And up till today, I have not heard that. I'm still hearing that. For you, I gave up parliament job and I'm working with you and doing my own dishes and doing my own clothes. So I said, hey, it was voluntary. Anyway, she got an MBA in marketing and finance at Rutgers, got a program of instruction from lawyers from Harvard Business School, was promoted very quickly to controller in Xerox Corporation. So between the two of us, we pretty much covered both the technical area as well as the finance area. And so if you want to get into a good, solid tech-based company, I think, in my opinion at least, you need to have good credentials that people will have a hard time questioning. Then she started her own business, Bauer Financials, and all small startups used to come to her. And she was called the Loan Arranger. So there was an article published in a Texas newspaper calling her the Loan Arranger rather than the take off on Lone Ranger. And then she co-founded Colorado Histoprep with me. And I said, I don't know anything about histopathology. And she said, you're a scientist, no? You will learn. And like a fool, I believed her. You know, they say, fools rush in where angels fear to tread. This was true. So I rushed in, and I'm so glad I did actually. Okay. Well, it's true. As chemical engineers, we can learn anything. So I picked up this stuff, and pretty soon I was applying chemical engineering principles to it and making superior products. So we founded Colorado Histoprep in 1999. And this is a company which basically provides histopathology services to the life sciences companies and researchers. Now, we were turning around the tissue slides in the readings so quickly that some of our clients told us, why don't you add hematology and clinical chemistry? Because hematology and clinical chemistry, really, you get the results in a couple of days, okay? And these people were basically making them wait for months to get the results. And our reports were going in faster than the hematology and clinical chemistry services. So we started that. And so including histopathology, hematology, clinical chemistry, and then we did this for GLP as well as non-GLP. Now, what were some of the startup challenges that we faced in Colorado Histoprep? Funding, okay? Basically, when you try to open a business, at least in the US, I know, they say, okay, how are you going to fund it? So we put our house on a second mortgage and we took a line of credit against the house. So that means basically if it fails, you have no place to live. But we did not fail. Then there's a conservative bias against foreigners. You're brown people, you don't want to do business with you. How do we know you're smart? There's still some of that, less so in the US than in some of the countries like UK perhaps. I only speak secondhand about this. All I know is that there's a subtle bias. Okay, they won't say no outright, but they won't say yes. And so we had to overcome that. Now there was a resistance by the seller. This was really very interesting. Okay, because we were financed by the bank, but the seller did everything that she could to try and make us fail. I think her hope was that it fails and she gets the business back and runs it. But you know what? We weren't going to fail. So here's what she did. She picked up the phone and she called the fire department. And we got a surprise visit from the fire department. And one of her friends was there in the fire department. So she went around the lab finding all the places that were a problem and had been a problem for 10, 15 years. Okay. Got to the point when it was so ridiculous that the other people who were with her, I said, what are we supposed to do about this? We were here 10 years ago. And the other people said, yeah, what is he supposed to do about it? So I think that helped in that it was so obvious that she was setting us up that basically that person got overruled. Okay. Then we had a surprise visit by OSHA. Again, she called up OSHA. And OSHA came and audited us. And guess what? We passed with flying colors. Okay. Next, we got audited by the FDA. So all these, I don't know about the FDA because the FDA doesn't come if somebody gives them a call, but they just came because we had changed hands, no? But the first two, definitely she played a role in calling them and trying to bust us up. But didn't happen. We survived and we thrived. Now, what happened then was we had various clients for Colorado Histoprep, which was basically a histopathology company and analytical chemistry and hematology company. So what we did was we decided that this is too small a portion of the pie and we wanted to expand it. So we acquired Care Research, which was a client. Okay. And this you can consider as a vertical integration because we provided the services after the animal tissues were harvested. And Care Research was basically a client who was doing the in vivo toxicology preclinical services. So we acquired them in 2010, and that's considered. You can consider that as a vertical integration. And what we do now together? Care Research and Colorado Histoprep provide preclinical testing services to pharmaceutical companies, biotech companies, device companies, university researchers, individual researchers. So basically, pretty much we have control of the entire process. We basically have, you can say, practically from soup to nuts. What we don't provide at this point is analytical chemistry services, which we have to outsource. So basically, what the clients are looking for, what the sponsors are looking for, okay, if they don't want to hassle themselves with placing different parts of the project at different companies, okay, they want to have one point of contact where they say, okay, you take it, you make it happen. Because the non-GLP reports are not subject to submission or inspection by the FDA. And so pretty much the only time you want to go into a GLP study is if you pretty much know what the results are going to be beforehand.
Okay.
Does that make sense?
Yes. Yes, sir.
We try to tell that to all our sponsors, but not everybody listens. A lot of them want to jump in and do the GLP study. And we're telling them, Baba, if you get bad results, we have to report those. And then it's like walking in between the raindrops. You get results which you don't want, and you have to report those. You're done, basically. So we have been through this road many times. Where the client will not listen. They want to jump into the GLP study right away, and we try to advise them. But you can only take a horse to water. You can't make him drink.
Thank you, Rajan sir, for the energetic presentation, and thanks for the video. I think that would really helps some of the aspirants who wants to start their business in the USA. We know that's other side of the coin for them, but it's a kind of giving heads up to the fresh aspirants who comes out from the BITS Pilani or some of them. I have one quick question on the services, animal services, sir. As you mentioned that it is both GLP and non-GLP that you guys offer for the. Is that for a preclinical end-to-end solutions do you offer, sir? Is that anything specific window or specific services that you offer, or is that complete end-to-end solution for the preclinical part?
Okay, so like I said, as far as are you talking about the nature of the compound, or are you talking about the nature of the service?
Nature of service.
Okay, so the nature of the service, we'll do the preclinical part where we'll do the in vivo testing, and then we also will do the histopathology and clinical chemistry. Or if you have a preference for the histopathology lab, we can send it to that lab. Or if you have preference for the analytical chemistry lab, you can send it to that lab. Now the dose formulation, analysis, etc., we don't currently do that in-house. So we have two choices. One thing, we can recommend a lab that we have got good experience working and they give us good results. Or very often the sponsor might have somebody that they're already working with that they would prefer us to send the samples to.
CD3 Zeta. And so this 41BB, these two sections this generation of CAR T is the second generation of CAR T. The first generation didn't have what they call a co-stimulatory domain. It only had one signaling domain. And now there's actually up to 5 different generations of CAR T. And the CAR Ts that we see on the market today are the second generation CAR Ts. And there are 5 available CAR Ts and commercially available in the United States. And they're all second generation. Now they have different co-stimulatory domains, they have different nuances, but essentially they're all the second generation CAR Ts. Now, you probably have seen something quite similar. It really highlights the process at a high level of what a CAR T cell is. And so I took this off the Dana-Farber website. And essentially what you have, and it's pretty much the same for all CAR Ts, is you have your leukapheresis. That's your first part, where you collect the patient's T cells. And then you send them to the manufacturer, and then there's 3 major steps there, which is the isolation and the activation of the T cells. Then you re-engineer those T cells, and then you finally grow and expand them. And you have to appreciate that that whole process takes a significant amount of time. And then finally, the cells get shipped back and they're infused to the patient. Right now, most all CAR Ts available are autologous of nature, which means that you're taking their own patient's T cells and re-engineering their own T cells. Now, there's a lot of research around allogeneic CAR Ts, and those are really taking other T cells from healthy donors and re-engineering those, but none of them are on the market so far. So what does the personal supply chain therapy imply? Well, first, it's a multi-step process. And so this is pretty high level, but essentially, the first thing you have to do obviously is enroll a patient for CAR T. And so the patient's got to be referred to the hospital, and then you have to have a treating staff to enroll them. That at that hospital, and that's done at the hospital. Then they do what they call apheresis, which is taking the patient's T cells are collected, and then what happens is cells are either collected fresh or cryopreserved, and then they're shipped to the manufacturing facility, where, like as we mentioned earlier, it goes through three phases. And then after all those phases are done, then the cells are frozen and there's a QC release, a quality control release. Once that's done, then they're shipped to the treatment center where the patient can receive their cells. And then there's a whole procedure around post-treatment follow-ups. Now, if you read more about it, there's this concept called vein-to-vein time, which is really from the apheresis aspect to when they receive treatment. And the vein-to-vein time is important because it really dictates how much time what's the time it takes to actually receive the product. And so right now it's between, I guess, anywhere between 3 to 5, 6 weeks is pretty much the vein-to-vein time. It's important to appreciate.
Hello. So when you say
Yes.
the product, should I take it as a lentiviral product?
Yeah, well, the lentiviral product is part of it, right? And so the lentiviral product is the full product, right? They add the viral vector in the manufacturing process. So you take your T cells and then they re-engineer and put a viral vector attached to it, and then that's so from the moment the patient gets apheresed to when the full CAR T is injected is at the treatment. So that period is the vein-to-vein time. Does that answer your question?
Yes, you can proceed.
All right, thanks. And so that time, so that takes anywhere between 3 to 6 weeks, and that's important because there's some therapies out there, some diseases that are very acute, and sometimes patients don't necessarily have that time, and so there's different I guess, treatment options that physicians do to see if the patient can, make it to receive their treatment. And so that's the multi-step process. It comes back cryopreserved for all the CAR Ts, and at the site there's a thawing process, and then it would be injected there. When we look at the implications of the CAR T at the hospital and the manufacturing site, these are just examples, but you can certainly appreciate from a hospital perspective, there's a lot of different steps involved. In the first, obviously, patient identification, then ordering the product, and then in the ordering, it has to be aligned with the apheresis, and everything has to be coordinated, because if you take the manufacturing is really done for that patient, and if that slot is reserved for the patient, but you don't have it, you don't have the raw material, then that slot is gone, right? And so everything is really coordinated. And so the ordering is aligned with the apheresis schedule in the hospital, and then that goes through the process there. Then the hospital needs to ship it. Everything has to do with what they call a COD, with chain of identity. So every product in that, it seems quite simple, to think that labeling a product is easy, but in reality, it becomes challenging in the sense that you always have to know where the cells are, and they always have to be attributed to that patient. It would be, a disaster if the patient didn't have the right cells back. And so there's a lot of human steps involved. And so then they're sent to the manufacturing, then the center waits for them, and then there's the whole delivery that comes back, and they have to, unfreeze the cells, prepare, administer, monitor, and long-term follow-up. So that's from a hospital perspective. When we look at what, these are examples from a manufacturing, you can easily understand that, there's the whole, and there's just examples I've put down below, but of, what it means for them, from the apheresis pick-up, the collection, the identification, and then really monitoring where the cells are, planning the whole site management and communication, there's constant communication, delivery, organizing the courier. It's not a typical courier that delivers that, and you've got fresh cells, they're traveling, and so there's that, and so there's a lot of stuff that's happening behind the scenes. And my point is that there's a lot of steps, and on both ends, it's a lot of coordination. And so what's done from a commercial readiness perspective is when you set up a clinical or a commercial hospital site, there's, as you can appreciate, there's a whole readiness procedure, which really looks at, making sure the apheresis team is trained and qualified for this product. And there's a whole list of SOPs that are in place at the hospitals that need to be done, and specific training around that. Just around the infusion and the product handling aspect, there is that aspect, the enrollment and scheduling and the order flow, what does that look like? And there's the whole, from when the product's commercialized, but there's a whole risk minimization certification around that. And it's really meant to protect patient safety and put a process in place that allows to manage your long-term effect, and have, also specific training on drug-related adverse events like you'd have with other compounds. And so, and from a commercial perspective, there's another layer that makes it also, that's important to address, is there's the whole contracting aspect, which you can appreciate that contracting is, has, there's a whole bunch of ethical questions that are part of this contracting, like once the cells are collected.
Can you brief on product-specific training? Because
excuse me.
Can you brief on product-specific training?
Yes, so of course, and so the product-specific training is really related to the product itself, and so more around AE toxic side effect management, and so that are related to that product. And so, and if there's any specific training around, when the patient has to come in back to the hospital, if the patient has to be hospitalized, how long should they monitor for specific adverse events and whatnot. And so that's the product specific training. Does that answer your question?
Yes, please proceed.
Great. And so I was thinking from the contracting, there's ethical questions as well that have to be addressed on a commercial front, because who owns the cells once they're shipped? Do they belong to the patient? And what happens if product doesn't come back within specification? When you do the whole process of manufacturing, they have to be what we call on spec, and within the number of cells that are within your regulatory approval. If they're not, then what happens then? And so that all has to be clear on the contracting, and so the hospital has to sign that, and so it's a nuance on that. But the point is, there's a lot of preparation that is done upstream to get a hospital ready and get that done. And so it brings us to the topic, which was the topic of today, and so what are the current challenges of commercializing CAR T cells? But I don't address India per se here. But what I see is, this is going to be evolving in the future, and so what we have, like what we're seeing right now, and we'll link in India as well in this kind of possible solution, because I see countries like India having all countries, being able to, as this becomes more efficient and changes the scalability model. My point about the presentation today is the scalability is the limiting factor, and it costs a lot to produce them. And that's another limiting factor for many countries. And so it's only a small group of countries that really have access fully to these products. And so the possible, so what I see is
can we know that small group of countries? Can you name them?
The countries that have access. Yeah, of course. So every pharmaceutical company has its own different, depending on how they evolve, but typically countries that have access right now to commercial CAR Ts. Well, actually, I shouldn't put all pharmaceutical companies in the same range. It really depends on where they are in their growth cycle. Okay, when they start off, they can only meet demand, and they've got the centralized model typically in the United States, and they can only meet demand there. So they start with that country, and then they go to Europe, and whereas then they build a manufacturing plant in Europe and start serving that. And sometimes it's done in the same sequence. And then you've got a few countries in Europe that have a system that allows to pay for these products early on. We have to, a lot of these countries take a lot of time to get these all cancer treatments in the system and get paid. And so countries that have a system in place typically are France, Germany, Switzerland, and also Japan has a model that allows them to be able to fund these products earlier. And so that's how that goes. And then there's a next wave, and that's where we see that of countries. So that's kind of how that tends to be globally expanded. And it's all about being able to supply. And you've got other, you've just read in the news not too long ago that, there's another big expansion phase for the Kite Gilead products. And I forget which other countries they were serving, but it was a nice big extension. So that really creates a lot of hope as how this evolves. And it's really linked with this this personalized supply chain, right? And so, so bringing it back, I guess, to the possible solution and what are the future trends. So there's these semi-automated manufacturing process, which you can appreciate that if you have, less human process and you've got semi, then you decrease your costs. You also you more reliable, right? Less chance of human error. So there's that aspect. Then there's the fully automated manufacturing that we're seeing. There's these technologies that are completely full automated that we're waiting. Oh, sorry, I'm going back to the semi-automated. We expect a commercial product within about 2 years. There's one that's quite advanced and so we might see this in the near future. The fully automated, there's a couple of technologies out there from centralized, fully automated, but there's another model that's really neat. It's called more of a cocoon, where it's done in the hospital at the bedside, and it's all produced there. So you can, you remove all the shipping and process. So there's a lot of evolution on that front that's moving along, and I think that's going to also help more countries have access to these products. The other is what we're seeing is we've got one company that actually all the companies that have products out there, what we're seeing is they're working on decreasing the manufacturing time. And so by increasing that efficiency, you can run more of these patients with the same manufacturing facility. And so they're all decreased. And so there's that. I'm really encouraged by one company that I saw and actually has their first phase 1 trial in patients done, where they've got a 24-hour manufacturing time. Now, remember that typically it's 14 to 21 day, just easy. Actually, it's more about a month in the manufacturing process by the time you isolate, you engineer, and you grow. Well, this company's done that in 24 hours. And then they have a 5-day QC release. So if you can have that, then you've just increased your production by a lot. And so there's all kinds of strategies that are coming into play to make these CAR-Ts more, scalable. And then there's we haven't touched on allogeneic CAR-Ts. They bring a lot of hope. I'm not sure if they're the full solution, but there's a lot of interesting data. There's a couple companies that are in the runnings that maybe we might see a first allogeneic CAR-T again within the next 2 years or so, commercially available. And so what the allogeneic CAR-T would do is you would take T cells from a healthy donor, and so you can produce large batches and you can have them on the shelf. And so when the patient comes, you could use it as you would do with either medicine. And so, and then there's all kinds of really cool stuff with NK cells, even macrophages that are being studied. So I'm quite hopeful that many countries will have readily accessible CAR-Ts in the future. Not today. It takes time, but I'm very hopeful that these are going to be great products for many, and hopefully help more patients, right? And I guess this is my final slide. And so, I think if there's one thing we have from all this, that it's complicated, it's expensive to make, it's risk. There's a lot of risk here. So why do you pursue CAR-T? And I want to finish on this because at the end of the day, this is the first patient ever receive a CAR-T. Her name is Emily Whitehead. I think she is the poster child of CAR-T. And so her story is about, having an acute lymphocytic leukemia at the age of 6, and so ALL, for those who don't know, when you can treat it in a front line and cure patients. But if you relapse from that, your chances of survival are, about 30%. And so at 6 years old, she had relapsed from her first line of therapy, and her chances were 30% survival. And that was the first CAR-T patient. And this year, she's just celebrate 10-year of cancer-free. And so I guess that's why it's so promising, and that's why we do it, and that's why there's so much excitement around this modality and technology, because certainly what we're seeing is that it can help a lot of patients, and hopefully help more patients around the world as we become better at scaling and commercializing and bringing these products to market. So I'll stop here. If there's questions or comments or.
Yeah, thank you for the presentation, and thank you for giving us insights on commercialization aspects, and giving us insights on how scalability is still an issue in different countries. So panelists and participants, you have any questions for Guillaume?
I just wanted to know, see, major problem is two. One is, we know that, the gene therapy was not a great success. It is a kind of mixture of gene therapy, along with that, purification, GFP facility, even the depletion of the B cells sometimes, what I read. I'm not a person who has worked on this, but I am more a molecular biologist and immunologist. So I understand the structure, this and that, or the PCR and BCR. I worked at MIT for a long time of that. So how do you think it will be kind of visible down the line in terms of the money? That is number one. Second is investment. It's not about one patient is giving and it will be engineered and then it will be reinfused. So the point is, it requires a huge facility to run. So how do you think that, unless even, I don't know how many people can really afford, and I don't know about the insurance. So if you can highlight on that. This was a discussion, but I thought we may need to know more what might happen in a country like India down the line.
So it is really. So I just want to make sure I get the question is really around the high cost and how can a country like India be able to think of producing, making this readily available and producing it in your country? Is that the essence of the question? Yeah. Well, I mean, that is a big question. What we're seeing, and that's not the solution, but I'll show you what we're seeing is we're seeing many researchers doing their own clinical trial at a cost that's initially what we like is they do their own CAR T and they do their own clinical trial and they're advancing that. That said, that eventually, when it's one thing to run a phase 1 clinical trial, but the moment you start building a bigger clinical trial and you want it to get to more patients, the cost of will increase quickly, and then there's a lot of specification. One country that I've been following that's really a world leader in CAR T is China, and the Chinese have done a lot of really fascinating science, and they're advancing this quickly by they've got a strong group of researchers, and so we're seeing them do a lot in that front. And obviously, from a Chinese perspective, their efficiency has been, I'm not sure if we're like, I'm monitoring, I follow their data and I follow them. I also know that they can produce a CAR T for a fraction of the price because of the way the system is set up, and they've
the
got
major issue. What I know is the T cells are not very good for any kind of transfection or viral mediated entry of any genes. T cell is I worked on T cells, and the whole world knows. have to take the T cell and put engineer a new gene, that itself is one of the biggest challenges. Because T cell proliferation also require a lot of CD3, many other activator, even to proliferate. And after the engineering, get a not even near to 100% of the cells to have this transfection done, it's not very easy. That is one of the biggest challenge. Now, let's say if it is 20 or 30% transduction rather, by the virus or the lentivirus. The rest of the 70% will be the cell or autologous cell from the patient, which will again go back to the body. So that is another major challenge, because there is, I don't know if people are purifying those engineered cell by some way, because there is no GFP or any other markers or to do the fact sort of those. So that is another thing. I don't know how to solve that. Like in HIV, people thought, and other genetic disease, people thought, but there and here there is a difference, because here each lymphocyte cells are basically cancerous cell from a patient, if I got it correct. So 30% of that, if it is transduction is done by lentivirus, and 70% again go back, whether they will be creating the same problem that it was doing without engineering.
There's not, from my perspective, I mean, that's that's not something that I can, I don't have an answer for you, to be quite honest, about at that level of, how do you expand it and create that aspect, my expertise is really, once you've got a product available, is how to, how do you set it up commercially, and so.
See, in other genetic disease, if you give the new cells, instead of taking off all the T or B cells and giving it a fresh, even if you give some cells, they will start making the new protein, which are corrected proteins, right? So the gene therapy can partially even work otherwise for this genetic disease. But here, lymphocytic cells, they are all around. Taking that off 100%, not very easy. Gain all the engineered cell 100%, that is also toughest challenge. So these are the things also and we are playing with the immune system. Apart from that, there are a lot of side effects, as you know, the cytokine storm and all. So those are also needed to be really thought of before we really invest or really put a lot of effort to. But definitely it is one of the best mechanism and probably possible mechanism to handle this type of cancer.
Yeah, no, I think, and so you're doing a lot of work right now on T cells and working on.
And not really. I was working, but right now I'm not working on that. But my research was on T cells for 20 years or so. And before that, I was at MIT. So there a lot of people was working, including a lot of Nobel laureates. So I kind of know the field, but these are very good thing to do. In fact, I also started purifying the stem cells. Pluripotent stem cell probably was a good thing, but that is also not very well known, whether CD34 is the only marker. There are many other complications that has come even for the pluripotent stem cells. So that's another biggest challenge. And gene therapy, that it was kind of failure in many cases, although there are some success in some cases. This is probably always we love to hear that 6-month life has, gone up to 2 years or 3 years. We love to hear that. But in lieu of that, the amount of money, the cost, the investment, whether that will ultimately permit or not, that is the question. That is one of the biggest questions. All of us know.
It's the question not only for India, it's a question for the majority of countries, and not just that. All gene therapies, what we're seeing right now is they're coming up with extremely high price tags, and you're wondering how is that going to work, right? And are we going to have access to these gene and cell therapies? So that's a question that's on everybody's mind.
One more
And-
comment probably is that, in between the last probably 10 to 15 years, there are a lot of David Baltimore MAGE therapy. There are a lot of peptide-based therapy was available, which also partially worked very well, but later on they gave up. So if it can be a combination of those peptide-based immunotherapy plus the CAR T cell, probably that is not a very bad idea, because peptide can access most of the cells without having infusion and all this, taking it out and then engineering. So I was thinking that, why not try a dual system in which give the peptide as well as this lentiviral based CAR T cell, so that, it can work even better.
There's a lot of, that's actually a great idea, and I've seen all kinds of literature as well that they're looking at different combination to activate the T cells as well as part of it. And so there's just so much going on in this space that's touching on so many different aspects, right? And I mean, it is fascinating to follow where all this is going, and there is so much research, that's done worldwide on cell and gene therapy, and right now that, I mean, there's going to have an impact for sure. And there's also I should have read about that. I remember reading a headline that there was one Indian company, a CAR T company, that was looking at producing a lower cost CAR T. And I should have thought of researching it.
I was also listening.
Could
I
be
was also
IIT
reading something.
working with TMC.
But going to abroad and getting it done almost for 10 times.
Yeah.
I have one minor question, is that why there are so much of cytokine storm, you think? Because unless there is a non-specific crosstalk, and that allows many of the cells to go to the inflammatory pathway, I don't find much of the reason why there has to be, IL-6 and many other cytokines. They just shoot up many cases. Any idea?
Yeah, there's all kinds of, I guess, well, they don't all produce all the same level of cytokine storms, right? And so you've got some agents that I've noticed that don't produce as much, and some of it is just by the mechanism of how, well, there's all kinds of theories which aren't validated, right? And so you've got a CAR T that doesn't produce, and then you're looking at, okay, well, why don't they produce that many CRS, cytokine release syndrome? And then there's all kinds of theories as to why that product might not have as many CRS. And sometimes it's about how they've designed the product. Sometimes there's one that's humanized, and they think it's maybe that. And so I don't think there's nothing really concrete, or no one will say officially that that's a reason. But I think it's more around the construct as to why there's some that are produced more than others. That's kind of how I'm understanding that.
The CD19 is another very important major issue, because many other cells like heart cells, many other cells also produce a lot of CD19. So CD19, after that, a lot of people actually get major issue in the heart attack and others. So
there's a lot of CD19. There's a lot of different CD19s out there that are being researched on, and so that's obviously a major one. There's a lot of other. There's a whole bunch of different. You see CD19, CD20s. There's a BCMA CD19. There's these bispecific CAR Ts. There's, yes. I mean, there's so much. And then they looked at whether it's armored or not, and I mean, and then we're even seeing the first. I saw it the last conference at American Society of Oncology, the first solid tumor CAR T that's moving into phase 2. So there's that. and they're different, targets. And so that's just for cancer, but you're seeing a lot of early, interest in other non-cancer diseases. And so I think this technology is really being studied in a vast indications and possible diseases. So I think we're really at the beginning, although it seems like, the first CAR T approved was in 2017 in the United States. So it's 5 years now, but I still think we're at the early beginning because there's so much investment done from a research and development perspective, and there's so much on that front that it's really the tip of the iceberg. And so the question is how can we bring this therapy to more patients and worldwide, and how can more countries have access, and how can we decrease cost, and so I think that's the challenge.
Okay, thank you. Thanks for a wonderful talk.
Thank you. And thank you for your comments and feedback. Appreciate that.
So I think today's talk is kind of relevant to our knowledge on that. But as you know, it was a big failure. And the failure was because of many reasons. I quoted that just because to tune you to say something about how our immune system is built up, what the immune system actually sees in our body. When a pathogen comes, what happens? And today's discussion is a little bit different because here there is no foreign material, there is no bacteria, there is no virus, but it is our own cells, right? We have 2 billions of cells. Out of that, in the blood, we have many kind of cells. And now we have generated a new kind of cell, which is also called the cancer cell, which is not very easy to eradicate because it is inbuilt, it is already there. It is not easy to find it out, and because not lot many receptors are there which are on the surface of the cells, it is not very easy to get rid of that. And if it is a solid tumor, there are problems. If it is a leukemic patient, there are other problems. So with that, people thought for last 30 years or so that what can we do to make sure you train a particular type of cell, that is T cell, in such a way, just like a dog, those sniffer dog that we call. So all that they do, they go to and allow them to smell the gun powders, those kind of things. And these dogs are specialized for a particular smell. Okay, so they have a receptor, and the receptor work for this gunpowder or metals. Okay, then they take them to the flights. If there is a problem, these dogs actually can go and these are called the sniffer dog, and they can sniff, and then they can find where the problem could be. So similar thing we wanted to do in the case of the cancer. So the idea is that T cell is the one, and the B cell, they actually understand one thing, and that is which one is the foreign and which one is the cell. Okay? So because of that, they can actually find out very quickly if there is any abnormal cells. Now, these abnormal cells in this case is the cancer cell. So we want to take the T cell out, engineer in such a way that it will have a receptor which will specifically can get the smell of a cancer cell. That means some proteins, some receptors which are on the cancer cells, they can find them out. And T cell has a property to kill those cells in which it can interact very tightly. It is always interacting with other cells, but our own cells are never killed because the interaction is different. But if the interaction is a foreign receptor or a bacterial peptide or a viral peptide, then it will either engulf or it will kill. So that is the problem. So we are going to use the T cells and give some property in such a way that they can kill the cancer cell. And this is also very well known that cancer cell has a very extra property, and that property is to hide or it can escape our immune system. Because these cells are our own cells, so they have a much better advantage to hide. Okay, so this is like what I think is a friend is not a friend. So that friend, when the time comes, can put me knife. It is like that. So the bone marrow makes all the blood cells. You all know it, T and B cells, and they are the one which are called the immunosurveillance system. The immunosurveillance system is not only T and B cell, but there are macrophage, there are NK cell, there are many other cells together which are present in the blood. Together, they functions to eradicate all the foreign particles, foreign bacteria, etc. And this is most commonly, especially in the lymphomas, in addition to other cancers as well. So they can change the immune system. They can change the immune system cells such that once perfected your body, it begin to interfere with the normal cells, the normal way your immune system works. Cancer cell can get into the bone marrow cells, and once it is metastasized or once it is going somewhere else, it is not very easy to eradicate, especially if it goes to brain. Although we have a blood-brain barrier, but sometimes these metastatic cell gain some properties by which it can even go into the brain. And once it is in the brain, it's not easy to take it out, because majority of the drugs does not work against any cancer, including the glioblastomas. So this is a cartoon showing that the T cell is interacting with other cells, and it has a property to kill other cells which have either foreign peptides or as other receptors. And that is how the T cells has this extra property to crosstalk with many other cells, including the cancer cells. And thus, T cell can be useful to generate this engineering in such a way that it can specifically interact with the cancer cells even better. So the T cell, for this purpose, what one has to think of is that T cell get activated. T cells are one of the most smallest cell produced from the bone marrow. Then it goes to the thymus. In the thymus, it gets educated and come to the blood back, right? These are very small cells, round cells, very tough cells also, and not very easy to engineer. That is another problem. I was asking this question in the previous speaker. So even lentivirus or any other things, it is not very easy at all. People started that from gene therapy to HIV to many things, but most of these are failure because efficiency of putting any gene inside a T cell is another biggest challenge. However, this rarely occurs. What it is saying that normally the T cell will target via the antigenic sequence presented on the T cell receptor via MHC. So MHC is the molecule on which the bacterial or viral peptide will sit, and T cell will come and find this structure, and it will bind, and then it will start killing it, okay, or engulf it for the macrophage. So this is the way T cell works, okay. So there are different kind of the T cells, T helper cells, and then other T cells. Within the T helper cell, there are Th1, Th2, and then other cells like Tregs, Th9, all these sorts of T cells are there, and each of them has a different kind of functions. Now, T cells on the surface wanted to put, we thought that, meaning the scientists thought that we will put a very important chimeric antigen receptor. What does that mean? It means chimeric, you all know. Chimera means it's a mixture of different characters. So you're making that, engineering that, cloning that, doing the sequence, and putting that back in the laboratory, and then you are putting it back to the T cells. So that is why instead of the T cell its own receptor molecule, it will be expressing a modified T cell receptor, which is also known as the chimeric antigen receptor. So in this case, antigen should be known, and that is present on the surface of the cancer cell. So you are designing the antigen receptor of this chimeric molecule in such a way that it will specifically interact with a malignant cell, which is on the surface of that cancer cell. So its own receptor is engineered in such a way that it can only interact with the cancer cell, but not the other cell. That is the whole purpose. And so for doing that, there are few things that also we need to keep in the mind. And that is that we have to take the T cells out, engineer it, and put it back after engineering and after putting the genes up. So now, for a long time, we can go back up to 1863. Okay, description of immune infiltrate to the tumors. So it has started, this idea is not today's idea, it has started long time back. Okay, and at different times, this immunomodulator, how to change the immune system, how to change the immune system such that will specifically do one function. Okay? So during the time, at different times, a lot of people have worked on this cancer immunosurveillance. People have given hypotheses, papers were published, wonderful papers published. And in 1991, after, almost about 37 plus 90. So after 130 years or so, people discovered the human tumor antigen. And this was going on, and some of them were approved, some of them were not approved to use. And by that time, this PT1 came, which was, 2014. And also before that, 2011, there is another molecule, CTLA4, on which there are, small molecules, and the antibody-based therapy started. Okay, so it is about 10 to 15 years from now, when definitely this antibody-based treatment started. And the latest one is the breakthrough status, that is the CAR T cell in DPIP. Now, if you look at here, this is the normal T cell. This is where my specialization comes, because I worked on the T cell receptor for many years. I was at MIT, and that time, I was working on the T cell receptor. So we were kind of the first to prove how the T cell receptors are being generated. How so many of the T cell receptors, which is 10 to the power 8 different T cell receptors, how they are being generated, although we have only 25,000 genes. So this was my research work. So based on that, it's very interesting for me also. So normally, if you look at very carefully, T cells has this, this T cell receptor, and there is a peptide MHC complex. The tumor cell will have this MHC class 1 peptide, and the T cell will come. Normally, in a normal case, it will not be doing anything. However, what we do is make a CAR T cell in which a kind of similar structure, protein structure after its three-dimensional structure would be looking like a T cell receptor, but it is not a T cell receptor at all. It is a totally engineered molecule, and I will come to that. So, and after you do that, there is not a easy way to put any gene to the T cell. So what we do, we use the viral vectors. In this case, it is the lentiviral vectors. This is a safe virus derived from HIV. It's a retrovirus, but it cannot replicate by its own. So it is a novel process by which it has this, it can interact with the CD4, and it will inject the gene of your interest into the T cells, right? So lentivirus is the one which is being used. So you can very quickly engineered lentivirus, a T cell receptor like molecule you can clone, and you can put that virus, that is a live virus, and the live virus will come in contact with the T cell and it will inject that piece of the gene, and that will be integrated inside. And once it is integrated in the chromosome, it will start expressing. Once it expresses, the antigen, which is shown here, will be on the surface. So this is our CAR T cell receptor. Okay. Now this will specifically interact with the tumor cell receptor and will kill. So it is a type of immunotherapy, adoptive cell immunotherapy. This technique allows the clinician to genetically reprogram the patient's own immune cells to find the attack and attack the cancer cells throughout the body. Then you have the CD28 yellow color, and then on the surface of that, on the top of that, you have made the T cell receptor, right? This is what we are seeing in the left-hand side. And now this is very interesting because this has every component. Instead of giving too many of proteins like CD3, especially CD28, you are making one molecule in which all of them are there. So it's a battery. You are giving a police with a pistol and some other equipments. So that if you give only stick, it will go, but it cannot kill, right? It has to kill. Now you have to give even sometimes such a weapon that it can kill even if the enemy is far. So thinking that, the scientist always thinking that how I can make my weapon the best possible for a cancer cell. So I'm saying the same thing, what I said so far. It will have a linker, it will have a variable region. The variable region will come together to make a receptor molecule on the surface. It is what we are following is like a structure like an antibody. Antibody can actually interact with any antigen, right? Any antigen. You have seen COVID. We have used this antigen specific recognition and immediate detection, right? So antibody concept has been taken. T cell receptor concept has been taken. So it is a hybrid of concept from antibody and the T cell receptor. So together we are putting that in a T cell to express it. Okay. So when we want, we can actually activate these T cells. So one thing I was asking this question about the cytokine storm. Many T cell respond to the target antigen. It requires the anti-inflammatory therapy. So whenever these patients are given, what they do, they immunosuppress. Otherwise, these patients cannot take these cells. Okay. So immunosuppression is very much critical. So that is another problem. And for B cell specific, the CD19 specific CAR T cell, if you want to think of, the whole of the B cell has to be taken out. Okay. So that is another problem. So you are basically tweaking with the immune system itself. Okay. There are technical and regulatory challenges. Who will take and who will not? It's not very easy. Producing the genetically modified CAR T cell. So who will take that responsibility? Even the gene therapy did not work. So using the virus, which is also a HIV-like virus, use that, and everywhere, if we depend on a company, हम लोग को लूट जाएंगे. This is another problem, right? So this imagination is good, thoughtful, it is very good, thought-provoking, but the point is reality. Can we ever be able to use such kind of therapy at all? Unless there is a quicker way. For example, one vector works for all. Number two, you can somehow see the problem is here that you have to take out the T cells. That is where the problem. That is also not a major issue, but that also requires a lot of money because this trapping is based on the antibody, right? CD4, CD8 based. And then using that culture that requires a GMP GLP facility. So that is another major drawback, I would say. So I think with that, I will finish my talk and I can take any question.
From the speakers from early morning, we are listening. So one query I have is one of the speaker told us that there is a scalability issue with CAR-T. So I think most of the speakers covered that too. So we come to know what is the issue with scalability. But there is another comment that we listened because you have been at different top levels in India. So what is that query is bioreactors. In India, bioreactors are not specifically designed for CAR-T. This was very technical aspect that the speaker has given from Deloitte. So bioreactors that we are using at every research institute is designed specifically for monoclonal antibody production rather than for a CAR-T or something. So what is your perspective on this aspect? Yeah, based on your
See, what we are talking here about is making the virus in truckload, right? We say truckload. So that is the biggest challenge, but that is not very hard to do. Basically, you need a facility where a lot of cells need to be grown, and once if you get the engineered vector means a company cells already designed the construct, which even you can get the lentivirus already designed or engineered already. So that is not very hard probably because it is a matter of doing a huge cell culture like Serum Institute. They do 500, 1,000 liters of culture at a time.
Right.
And the virus is made and replicated in a huge number because this lentivirus can grow very well. So that is not a major issue, I think. But the issues are more critical that the transduction efficiency, number one, the T cell to take those lentiviral construct and put that is the biggest challenge. And you have to remove all other viruses because you cannot put the virus inside the body. So you have to remove all the other lentiviruses. So basically, once the transduction is done, then you take these cells and culture it again for 2 weeks or so. But T cell has another problem. This is the problem of the T cell. They cannot keep on activated for a long time. Okay. So if you proliferate, there is a half-life of the T cell itself. It is not like any other cancer cells. Cancer cell can grow forever, but the T cell has a limit. After that, T cell dies. Okay. So even if you grow a lot, that doesn't mean that all of them will be live cells. If you don't give the live cell, there is no point because the T cell has to proliferate or interact with the antigen which is present on the surface of the cancer cells. So that is another problem, but that is not very hard. You don't need a bioreactor for that, but rather you need scalability can be done. So that is not a very hard issue, I think. Worldwide, there will be companies who can make the lentiviral construct. And once they make, the virus is easy to replicate, right? But the problem is, taking these T cells out, give the virus, and how much that will cost. That is another thing. So I think that is not a major issue at all.
Start my talk now. You know that microfluidic applications are, there in many areas of biological sciences, including point of care diagnostic, drug delivery. There are several analytical devices where microfluidics, plays an important role. So today, I would like to discuss about a few systems consistent with the open surface microfluidic platform as well as one closed system, and we will see that how this particular technology can be useful to increase several biological or biochemical functionalities. So why microfluidics is important, that just see these three examples I have picked up from internet. And you can see that we all know about this lotus leaf effect. And the second case, the rain is falling on the glass window. And the third one is we encounter in our daily life every day. But the most important part that I would like to convey through this particular slide is by just tuning the surface properties or surface chemistry, we can somehow, engineer the transportation of fluid, and which is very important in several biomedical areas. So from super hydrophobic surface like lotus leaf, we can go up to hydrophilic surface like glass slide, and also we have a surface where surface is very rough. So the surface engineering or surface characteristics, be it through chemical treatment, be it through some physical, through the changes of physical structure, we can bring control over the fluid motion. Now, as I was talking about microfluidics in biomedical applications, you know that nowadays there are several processes where we need to go for designing several artificial systems, like for bypass surgery need to go for artificial palm, and also if we need to go for some kind of drug delivery, and then we need to have a precise control of the flow so that that particular drug would be able to be there in a designated station for a while to do some to ensure that the reaction would be completed. So considering all this, I'd like to discuss one example wherein we can really have a control of the fluid, which is very important. So if you are focusing, say, the drug delivery, and if you'd like to deliver the drug, it may not be miscible with the blood, or even and it needs to be transported in a remote location. Then if we need to tune that particular flow with the flow which is there in a physiological system, and then to mimic the flow in a physiological system is not so easy, but at the same time we can really control by having some kind of grafting over the surfaces through which the fluid is flowing. So here that the first example that I will show that how if we consider that a particular slug or plug, if it contains kind of drug solution, if it need to be transported, and how it is possible to have a precise control that it can be transported in a judicial manner. So you can understand these are basically what it is done in the fluidic pathway. The surface or surfaces are decorated with pattern, chemically pattern, which may be having hydrophilic, hydrophobic, or oleophobic. So by properly tuning or adjusting that surface properties, we can really have a control, and you can see that that particular liquid can be transported in a very nicely or controlled manner. Next, I am coming to discuss about a few examples of the open surface microfluidics, which is indeed important to enhance mixing, evaporation, and separation. So why mixing is important? We all know that very often you need to go to pathological center for testing our blood or any other biofluids. And if we need to go for testing of our any blood or biofluids, we need to mix reagent with the sample collected therein. And there are several tests wherein if we can ensure that the mixing with the reagent can be completed in a reasonable amount of time, perhaps the prediction can be done very quickly. So considering that, we have done several experiments in our laboratory to see how mixing can be triggered in an efficient way so that the results can be predicted very quickly. So I'm not going to discuss this particular. So basically, to this end, we'll be discussing about droplet-based microfluidics. So if we collect a small droplet of blood or any other biofluid, and if we need to mix any particular reagent with that collected droplet, then how can we ensure that the mixing would be complete within a reasonable amount of time? If we do not do all these kind of things, it will mix, but it may take several hours. So keeping that in mind, our objective was to see how the mixing can be performed in a efficient way. What we did, we had collected one sample. So in this particular case, we had collected sample of basically having some ferroparticles, just to see that perhaps it can mimic the not rheology of the blood, but at least because you all know that we have hemoglobin. So basically, so you can see that we have taken very small droplet of volume 2 microliter, and that particular droplet contains a few iron particles, and we have used iron particles only to utilize the external fields like magnetic field, and we can see that that particular field can enhance the mixing. So what is done, if we look at the figure that is shown in the left panel, so this is the ferrofluid droplet which contains a few droplets. We could have done even using blood, but for that again we need to take some ethical clearance. So to avoid that, what we did, we have considered a small droplet, and if we need to mix another droplet with this the mother droplet, then how can we ensure that the mixing of this sister droplet with the mother droplets can be enhanced? And to do that, what we did, this is a typical configuration. We can see that the droplet is placed in between two magnets, and these magnets are of very small size, and these two magnets are energized or activated in a time periodic manner. So when left is on, right is remaining off, and vice versa. So if we have conducted this experience, and I would like to tell you that if we do not even perturb, if we go back to the previous slide, suppose we are not trying to perturb this particular droplet to be mixed with this sister droplet using this magnetic field, these two droplets will certainly mix because these two fluids are miscible, but it will take some finite amount of time.
What is the choice of electromagnet here, sir?
Yes. So basically, we have considered electromagnets. So because we had designed the circuit so that we can tune the time period of operation of a particular magnet, and when the right is remaining active, left is on, left is off, and when left is remaining active, right is remaining off.
So you purchased the electromagnet, or you have made the electromagnet? Pardon?
We had fabricated these electromagnets in our laboratory.
Oh, is it a micro-based or it is a larger size? What is the size of the electromagnet?
In this schematic, we had shown this in this way, but it is not very big in size.
Okay.
Discussing that perhaps in absence of magnet, these two fluids or two droplets will mix together, but it may take several hours to get homogeneous mixing. But if we take this particular arrangement, or if we keep this particular arrangement here, then we can accelerate the mixing process. So I told you that this particular frequency for this particular configuration, that is 3 Hz, is the optimum frequency for which mixing time is less and efficiency become maximum. So that depends on the volume, as I already said. Next is enhanced evaporation. We know that I'm not having expertise in kind of biological processes, but if we need to again ensure that the collected sample will be placed on a glass slide, and if we need to enhance the aqueous part of that particular sample very quickly, so that we can get some information about the residue. So how we can have enhanced evaporation? Again, the application of magnetic field could be an alternative. So issue is just one figure I am showing. We had considered a particular volume of the droplet, and you can understand that if we do not apply any magnetic field, that is the no magnet case, the total time of complete evaporation is of the order of 3000 seconds. Wherein just by applying magnetic field, and if we can vary the frequency, we can reduce the time, and you can see the total time is now remaining almost, total time is 1500. So it is now getting half as compared to the no magnet case. So this way we also can trigger that how a small droplet, if we consider of the order of the droplet volume is 1.5 to 2 microliter, and if we need to evaporate the aqueous part, how first we can evaporate, that is again an important in different biological processes. Keeping that in mind, we conducted this, and again let me show you these 3 movies, and you will see that here the particles are trying to go towards left when left magnet is on, and then particle again are moving toward right when right magnet is on. And there are certain physics, fluid physics, of course, I'm not going to discuss all those. That particular fluid physics is very important.
It's very interesting. But what is, to my surprise, how that is streamlined at the center, how the focused flow is achieved, and then it goes to the other edge. Obviously, the magnetic field is stronger, and then it is agglomerating. Like, how did you achieve that central, focused?
Yeah, so basically we had to go for, because the high-speed imaging, and we had to go for microfilm analysis only to focus, because there are 2 aspects. One is called bright field visualization, and another is called kind of microfilm analysis. So using high-speed camera, we could capture all these images, and then we had to post-process.
No, but there is no flow rate, right? There is no flow rate for this focus flow, because it's a droplet. It's just a droplet having the particles.
Yes.
And center, you can see it's a straight line. These particles go in a straight line.
Exactly. So basically, that is what, because when right magnets are getting, if we activate the magnetic circuit, and when left magnet is on, particle will experience this magnetic force and they will try to move towards the left magnet. When particles are moving, they are also dragging certain amount of liquid along with the particle, because that is the viscosity of the fluid which is responsible for this movement. And the movement of the particles is also not random, you can understand. So this is again due to the magnetic arrangement of the magnetic moment of inertia. So that is why I was telling that there are several physical aspects. So I'm not going to discuss all these things here, because it would be really, audience may feel bored, because those are basically purely fluid dynamics point of view.
So the choice of fluid here is water or?
DI water. It contains a few iron particles. If we go to the very higher frequency, you can understand that, see, the beauty of this particular analysis is that if we increase the applied magnetic field frequency, then that time scale of energization is very less, so that the particles are unable to move even from left to right. So in this way, if we compare this particular video with the video that we had seen few minutes back, in the last video we had seen that particles are going from left to right. Again, they are coming from right to left. But in this case, the particles are unable to move even to the right magnet. They are just oscillating between left magnet and intermediate space. So by tuning again this applied field strength, and of course the time scale to be precise, that totally modulate the fluid dynamics inside the droplet, and the overall evaporation rate is getting controlled. So this is what I wanted to show. Now if you go to the another experiment that we did, considering the this pandemic, so we had considered again a very small droplet of very small volume, 1 microliter, and that droplet is not the pure DI water droplet, but it contains 0.1% that sodium chloride salt, only to mimic the cupped and Sinis droplet properties. There are certain
sodium chloride. Can you repeat, sir?
Yeah, sodium chloride, I think 0.01, weight by weight, 0.1 molars. So this particular sodium chloride, we had to mix with the droplet only to get the. It is very difficult to represent or mimic the rheological properties of the cupped and Sinis droplet, but we had to consider that particular salt concentration in that with the DI water, so that we are getting closer to the characteristics of cupped and Sinis droplet. So this again, what we did, that you can understand even when there is a cupped and sneeze droplet on a human skin. So this is the saliva that my student took and placed on his own skin, and we had measured the contact angle. And when we place a small water droplet on the human skin, the contact angle is something else. And these two contact angles are very close. You can understand in one case it is saliva contact angle is 85 degree, whereas you know that for water it is 100 degree. Now, if we place, so it is very difficult to get saliva for the real experiments, but just to ensure that the contact angle is remaining more or less same, even with water in the human skin, we had considered that DI water with that 0.1 molar sodium chloride, that particular combination over the PDMS substrate, because we had considered glass substrate coated with PDMS layer, thin layer, and what we did, we had considered wind speed and the temperature to mimic the ambient condition here at IIT Guwahati. So we had seen that
the experimental setup is PIV, micro PIV.
Yes, it is micro PIV. Yes.
Was it built in-house or you have purchased it?
No, actually we have developed, because we have microscope, we have high-speed cameras, we have developed in our laboratory.
Okay.
So what we did, that as I said that the ambient, we wanted to see the evaporation kinetic in the presence of wind speed and temperature, and the wind speed and temperature that we had to consider here, considering the normal wind speed and temperature in our campus. So that typical wind speed we had considered is no wind speed, that is almost zero velocity, that is in the quiescent condition. And another two conditions we have considered.
My doubt is the micro PIV temperature based one? Is it a thermometry study that you are carrying on?
I didn't get your point.
No, the micro PIV what you have developed at laboratory.
Yes.
You can read out temperatures also, like thermometric studies can be done.
Yes. Arrangement is there. We can read out the temperature.
Okay.
And what we can see from here is that considering, if we consider the wind speed, when it is changing from 1.1 meter per second to 2 meter per second, we can see that the droplet evaporation is very fast. So that wind speed, not I can say that it is again non-trivially affect the droplet evaporation. That is quite common. We all know from our practical experience that if we place a droplet on an open surface and if we try to blow air with a high air speed, the evaporation will be faster. So even we wanted to see if the wind speed is normal wind speed that we already have in our room or laboratory, and considering that wind speed, we can see that that droplet evaporation is taking place even at a faster rate. So here it is the case where we could control the temperature, but you can understand that we had considered three different temperatures, and those are 298 Kelvin, 301 Kelvin, and 302, because we had to maintain a uniform temperature inside that particular experimental setup. So what we can understand that even for a change of 2 Kelvin to 4 Kelvin, the evaporation rate is really faster. You can understand the total time for evaporation, of course, the dimensionless time is 1 for the 298 Kelvin, whereas if we can maintain the temperature is 302 Kelvin, the droplet evaporation time is 0.6. That means, depending on the surface temperature on which a cupped and sneeze droplet is falling, the evaporation rate will be faster. And if we can somehow remove the evaporation aqueous part very quickly, the residue will be there, and that residue cannot be that much detrimental to spread that particular disease. I really do not know much about this, but the idea behind this experiment was like this, that let's see how fast we can evaporate the aqueous part, and not we, depending on the weather condition, a cupped and sneeze droplet, when it is falling on a human skin or any other surface, it can be evaporated. That the ambient condition can be favorable for the evaporation, depending on the location and of course the other issues. Now most important part is, that I would like to show you here that that if we consider a droplet of very small volume, and when there is a wind speed, that wind speed is not very high wind speed, say 1.1 meter per second to 2 meter per second. That is the typical wind speed in a normal condition. And when that particular wind is blowing over the droplet of such a small volume, then inside the flow field we could capture the flow field inside the droplet. And you know that if we consider, we had considered, particles of 0.8 micron. We could not consider particle of less than 0.8 micron. Our objective was to consider that if we could consider particles of 100 micron or even less than that, then it can mimic the real virus that would be there in a Kaplan series droplet. So the sole idea behind this particular task was to see how that internal flow field develops in the presence of wind blowing over a droplet, and that particular flow field can create a stress on the particle those are there inside the droplet. So I'd like to again show you two movies, and you can see that when wind is blowing over a droplet of very small volume of the order of 1 microliter, and if the droplet contains a few particles, how particles are getting activated inside the droplet. This is the particle motion, typical that we could capture again from microgravity analysis. Now, issue is
What
such a
is gray regime, sir? Gray or white?
basically because of some light we could not remove. So because we had to supply light from the bottom, and those are the basically kind of shadow that we could not remove.
Right.
So, but that is the movie we had captured from the top. So if we place a droplet like this, we had to pass light from the bottom, and we could capture that particular this particular plane is 50 micron from the bottom. And that if we consider this is a particular this is a typical small volume, we could consider to see what is the stress being applied on the particle because of this internal flow field. So that wind speed or wind when wind is blowing over the droplet volume, that particular wind speed creates or develops an internal motion. The motion inside the droplet, particles will try to have random movement when the particles are moving.
But at this time, there is no temperature. It is room
It
temperature.
is normal temperature. This is normal temperature you could consider. You can understand because we have considered 298 Kelvin. So basically the ambient temperature. So the normal temperature, that is the lab temperature, the temperature in our laboratory, and at that temperature we have considered wind speed and 1.1 meter per second. So you can understand when the wind speed is nil, even if you consider 290 Kelvin, the stress is very less, 1.62 micropascal. But when the wind speed is 1.1 meter per second, temperature is remaining the same, the stress which is orders higher than the previous case. So such a high stress can disrupt the viruses. So again, more in-depth analysis would be required definitely from the biophysical perspective to ascertain that the virus or viruses which are present inside the droplet won't be able to withstand such a high stress. So that part we could not do. Only what we could do is to see that particular wind velocity is developing an internal motion, and that particular motion is going to create or develop a stress on the particle. And if that stress the particle or virus cannot withstand, then the survival of the viruses would be a question. So again, I would like to show you another movie which you captured from the bottom. So you can see that this is the typical movement. So the movement of the particles is random. So they are moving in any direction. So next, I would like to tell you some examples of on-chip fluidic functionalities. So to this end, this is again a device that we have developed in our laboratory. Sole purpose was to create a droplet and to split the droplet in the same device. Because you know that if we somehow create a droplet, if the droplet is of any biological fluids, and if we can split the droplet in other end of that particular device into two parts, that two droplets can be taken for two different analyses. So that was the sole objective of this particular device, and that is the droplet splitter. Now, initially I'll be showing you a few results wherein this particular permanent magnet is kept, and this particular permanent magnet is helping to split the droplets of unequal volumes.
Disk magnet, sir. Disk magnet.
This is disk magnet, yes, small, very small.
Okay.
So the diameter is of the order of 8 millimeters, if I can recall correctly. And the sole purpose was that we can create one droplet in this particular T junction. That droplet will move further downstream, and here we have another T junction. And when that particular droplet is approaching another T junction, that droplet will get split into two parts. Now, depending on this particular magnetic field, we can split the mother droplets into two parts, but of different volumes. So I would like to again show you the movies. You can see that the droplet which is coming, and it is kind of, but here we have no magnet, but we have a small splitter, small, you can see the post, and that post is helping us to split the droplet into two equal parts. And this is basically, you can understand, this is symmetric splitting, because two droplets we are getting almost same volume. So mother droplet is coming and it's split into two equal volumes, and these two droplets are going into two divergence.
What is the diameter of that post? It is fabricated, no? You have made it in the mask.
It is fabricated.
So it is in micrometer or?
Yes. So total, it is like total height is 80 micron.
Okay, sir.
Total height is 80 micron.
And width is obviously 10
or 20 micron width.
20.
So and now, so this is the symmetric splitting, and this is again if we consider larger volume of the mother droplet. So by controlling the flow rate ratio, we can control the mother droplet that will produce in one T junction, and you can understand by having these two kind of figures that the droplet which is produced in the left, which is shown in the left panel, that volume is less, while the mother droplet volume is relatively higher for the higher flow rate. So again here I can show you this movie, and you can see that the mother droplets is getting split into two equal parts. So this is basically how that splitter, which is placed, or reed which is placed at the middle, can split the mother droplet into two equal parts. If we go to the asymmetrical splitting part, you know that now what I can do, we could do is just by placing this reed little offset from the center position, we can have asymmetric splitting. So here we are showing, that the droplet which is going towards top is less than the volume of the droplet which is going towards top branch is having less volume than the droplet which is coming towards the bottom branch. So this is basically now by adjusting the position of this particular reed, whether we are placing it little offset from the center. Here also you can see that again here you can clearly see the droplet volume which is going towards top branch is having less volume as compared to the droplet volume which is going towards the bottom branch. So basically by tuning the geometry of this microfluidic device, it is really possible to adjust the droplet volume that is very important in several biochemical processes, drug delivery application.
CARD stands for chimeric antigen receptor, that's CAR. And therefore, we need to understand how T cells are activated. I will touch briefly, then bring your attention to what T cells do for us in terms of immune surveillance against cancer and protect against viral infection in us. Then we'll go to some of the research that I did many years back, actually, when started my research career at University of Michigan College of Pharmacy. We were interested in developing a vaccine delivery system. At that time, there was no CAR T cell vaccines. Cancer immunotherapy was just in the developmental phase. So we thought that develop a vaccine delivery system using a liposome as a nanotechnology platform that will trigger cancer antigen specific T cell responses, that refers to T cell response. And what we did to achieve that, we got some help with a bacterial toxin harvested from Listeria monocytogenes, that is listeriolysin O, encapsulated within the liposomal encapsulation delivery, along with the tumor antigen, to demonstrate that, yes, if we do that, then we trigger a cytotoxic T lymphocyte response against tumor antigen, such as the melanoma model, and also extending our observation, we also could demonstrate that we could trigger viral antigen specific T cell responses. LCMV stands for lymphocytic poliomeningitis virus, is a mutant model of viral infection. The concept of vaccines to protect against infection, that was subsequently transferred in the domain of cancer immunotherapy or CAR T cell demonstrated. In the context of the vaccination, Edward Jenner in 1796 first demonstrated that pre-exposure to cowpox virus conferred protects against much more virulent smallpox infection. Louis Pasteur, the French scientist, about 100 years later demonstrated that altered microbial preparation can generate immunity against virulent pathogen. Those were the first scientific demonstration of vaccination. But as I mentioned, in India and China, ancient world, there are many literature that describe that concept of vaccination or variolation, etc. So question becomes how vaccines do really work. It's a complicated story on the left-hand side. Without going through all those things, if you look at two cell populations, one is a B cell, one is a T helper cells, and when we use a vaccine antigen, vaccination usually triggered good B cell responses. The green structure, imagine that's a vaccine antigen, and that a vaccine you can imagine COVID-19 vaccine. So for any vaccine, including COVID-19 vaccination, we triggered a tremendous level of very good antibody responses and T helper cell responses that secrete cytokine. These are CD4 T cells, but we hardly generate a cytotoxic T lymphocyte response, which is a CTL response, cytotoxic T lymphocyte. Why I'm mentioning that specifically in this platform? Because to treat cancer, to fight viral infection, we need to trigger cytotoxic T lymphocyte response. And when we heard about the CAR T cell platforms and etc., they all triggers or aims to generate cytotoxic T lymphocyte response. So little bit on the basics of the immune system. How do we generate immunity against any vaccines, any antigen? There is an innate and adaptive compartment, and T cell responses falls in the adaptive immunity, which is highly specific and generate immunological memory. So as I mentioned before, that current vaccination generates really good quality of adaptive humoral or antibody-mediated immune response, but very little or almost no T cell response. That's also pretty much true for current COVID-19 vaccines. And in our effort to develop vaccine protocol or vaccination cellular therapy to fight cancer. So some of the experiments we did to demonstrate that if we design a nanoparticle vaccine delivery system, such as liposomal vaccine delivery system, we can't generate tumor antigen specific immune response, but we can say that we can generate, but we have to assay, analyze, and demonstrate that yes, we have been able to generate not only that, but we can demonstrate that through cytotoxic T lymphocyte assay, ELISpot enzyme-linked immunosorbent T cell assay, and tetramer binding assay. I will explain this assay in the context, will not go into the detail. And of course, we also monitor antibody responses by analyzing enzyme-linked immunosorbent assay for detection of IgG class of antibodies. So what is a T cell vaccine? T cell, this is a complicated story. Don't need to go in detail, but since yesterday, perhaps many of you have learned about the CAR T cell, chimeric antigen receptor T cells. Very recent development in the fight against cancer is CAR T cell, cancer immunotherapy, but other sides of cancer immunotherapy or other players of cancer immunotherapy is immune checkpoint inhibitor drugs. These are the monoclonal antibody drugs targeting PD-1, programmed death 1, and programmed death receptor and its counterpart ligand 1 expressed in tumor cell that acts as an antigen presenting cell. And in the context of tumor-specific T cell activation, these molecules plays a very important role. Also, CD28 and B7 family of molecules, another pairs of molecules, they also play a very important role. What do they play? They actually shuts down T cell activation pathway, which is kind of okay when we talk about a viral infection-induced cytotoxic T cell activation, but that does not help a cancer patient to shut down T cell activation. So scientists created anti-PD-1 and anti-CTLA-4 monoclonal antibody drug that actually anti-PD-1 specific monoclonal antibody drug, Keytruda or nivolumab, won the Nobel Prize in 2018 for physiology and medicine. Along with PD-1 targeting antibody and anti-CTLA-4 cytotoxic T lymphocyte associated antigen 4 specific monoclonal antibody drugs synergistically, when they are used, that becomes the first line of therapy for many cancer patients these days, at least in US, particularly head and neck cancers, non-small cell lung cancer, and many other solid tumor cells. These days in US and elsewhere in the world, many cancer is first being treated with the cell-based therapy, cellular therapy, or immune therapy, rather than starting with cytotoxic drugs, radiation, and surgery. But these are the very recent development for last 4 to 6 years. About 15 years back, what we thought that why not we take a look at how we can trigger T cell responses. T cell refers to cytotoxic T cell that we can generate against tumor antigen, and then we can create a universal platform where we can bring melanoma specific antigen or lung cancer specific antigen or any other carcinoma specific antigen, then use that to trigger T cell response specific for that antigen. Our research hinged on that delivery system, and here let me review the delivery system, how we actually started working on that delivery system. So on the left-hand side, we have a CD4 T cell. On the right-hand side, we have a CD8 T cell. Here, we are mostly interested in CD8 T cell that fights tumor development, cancerous growth in our body, and that's also fights virus infected. Whereas CD4 T cells primarily gets activated to fight extracellular pathogens such as bacterial infection, whereas CD8's endogenous infection or intracellular infection and diseases such as viral infection or cancerous growth. But it is inherently tremendously difficult to create a vaccine formulation, vaccine delivery system to trigger tumor antigen or virus antigen specific CD8 T cell activation protocol. So how CD8 T cells actually gets activated? Cholesterol-dependent cytolysin, CDC family of toxins or proteins. Member is listerolysino, streptolysino, perfingolysino. They all have the ability to punch holes in the bacterial, in the cell membrane. That's essentially the property we wanted to harvest, utilize to create our vaccine delivery, T-cell vaccine delivery system. And here's a model of perfringolicin O PF4. These are the perfringolicin O, and upon binding to the cholesterol of the lipid bilayer membrane of any cell type that leads to the invagination of the bimembrane, then there is a hole is punched. That's what you see here. And essentially, listeriolysin O is secreted, released by the Listeria monocytogenes infection. And here it's in a double vesicular structure in the endosome. Then in the acidic environment, Listeria becomes activated, releases listeriolysin O or LLO that punches hole, creates hole, and that disrupts this endosomal compartment, and that releases replicating Listeria monocytogenes, then that is released into the cytosol, and then that invades into the next cell, cell-to-cell spread. We essentially harvested that hallmark or characteristics of LLO. So this hole punching, perforation of the double membrane structure is not limited to the bacterial toxins. We have actually those mechanisms inbuilt in our body, such as when complement gets activated, we generate membrane attack complex, and here are the holes punches through the membrane attack complex. Or when cytotoxic T lymphocytes gets activated, they also trigger 160 Armstrong diameter holes on its target cell. And who can be that target cell? A cancer cell, a virally infected cell. Ultimately, we punch holes through cytotoxic T lymphocyte activation, be it through a CAR T cell activation or be it through a protocol like what we attempted to do, trying to create a universal platform. Ultimately, through cytotoxic T lymphocytes, if we generate them against the tumor antigen, they will generate these type of holes, literally thousands of copies of holes, and that will ultimately kill, destroy a cancerous cell in the patient. So our strategy for T-cell activation against either tumor antigen or viral antigen employed cytosolic delivery of vaccine antigen in the MHC class 1 or HLA class 1 antigen presentation pathway, utilizing pore-forming listeriolysin O encapsulated inside a liposomal vehicle. So here's a typical liposome, and the lipid molecules can be very different, depends on the need. It can be used for transfection, it can be used to load up the cargo antigen to trigger many types of responses, including immune responses. And actually, there is a cytotoxic drug, doxorubicin, which has a liposomal formulation that extends the distribution and provides more efficacy in the treatment of the cancer in cancer patient population. So our strategy was to use phosphatidylethylamine and cholesterol hemisuccinate PECAMs in 2:1 ratio and encapsulate listeriolysin O in a very minute quantities. And as a model antigen, we began our work utilizing a very easily available ovalbumin to encapsulate within this liposomal environment, double lipid membrane. Subsequently, when we saw that OVA-specific antigen can be presented and triggering a T-cell response, cytotoxic T-cell response, we extended our observation to incorporate lymphocytic poliomeningitis virus-specific nucleoprotein. This is structural protein of a murine virus, and we also utilized another peptide, MHC class 1 peptide out of LCMV to demonstrate that viral antigen-specific immune responses we can really generate through this vaccine delivery system. So here is our cytosolic vaccine delivery system, which is pH-sensitive liposome that has PECAMs into 2:1 molar ratio and listeriolysin O purified from Listeria monocytogenes gene containing LLO containing gene that has been inserted into E. coli, and these are the vaccine antigen I already mentioned. We began our work with ovalbumin, subsequently extended to LCMV, and with liposome containing LLO, we can generate CD8 T cell response at a very efficient level, but without LLO, we still generate a CD8 T cell response because liposomes themselves are vaccine adjuvants and they can trigger some degree, not very efficient. tetramer binding assay, and this is the straight taken out of the flow cytometry, and this is the graphical representation of that data. So we can establish that enhancement of the viral antigen with liposomal LLO delivery system in that in a murine viral infection model, LCMV infection model. Filler data, while we targeted our approach on T-cell activation, it did not negate or abrogate antibody responses. You can see that LLO containing formulation and the vehicle containing formulation also triggered reasonable similar level of antibody responses. In summary, antibodies are much easier to trigger, whereas T-cell responses are not really very easy to trigger, particularly CTL responses. So this is, we have observed that yes, we can generate a strong higher level of cytotoxic T-cell responses when you incorporate listrolysine O into the nanoparticle delivery system, which is liposome. But then we ask the question, can we trigger a tumor antigen specific challenge response and protection, lethal tumor antigen specific protection, and LCMV specific challenge and protection? Yes, we have done that. I will finish up quickly. I know it's time coming up. So we immunize mice subcutaneously with OVA and LLO liposome twice, and with LCMV nucleoprotein specific antigen encapsulated in liposome containing listrolysine O, or without that. Then 7 to 15 days later, after twice immunization, mice were lethally challenged with melanoma. So here, let me take a pause. So this is 2 × 10⁵ number of B16 melanoma. This is a murine melanoma model we developed in the lab. That's not very difficult to do. And there is another cell line commercially available, B16 cell line, that is transfected with ovalbumin gene, meaning this melanoma cell line expresses OVA antigen on their surfaces. So that became our surrogate cancer antigen, what we can demonstrate whether we can generate OVA specific CTL response that will protect against this murine melanoma injected animals. And we monitor tumor progression and survival, immunized and subsequently lethal dose of B16 or B16 ovalbumin expressing melanoma cells in those mouses. In parallel, we also looked into lymphocytic choriomeningitis virus model after immunization with nucleoprotein of LCMV in LLO liposomal formulation. Have we generated adequate cytotoxic T lymphocyte responses? If we did, then it should protect those immunized animals if we infect them cerebrally, lymphocytic choriomeningitis virus infection. So let's take a look at the data. There are lots of lines. Without going into the detail, these are all controls. So MO4 was the murine cell line that expresses ovalbumin as a surrogate antigen, and we immunized this mouse with ovalbumin containing LLO or without LLO, then challenge them with respective tumor cell lines, OVA LLO, B16, or just without tumor antigen expressing, that's MO4. So if we look into this, and this is the control B16 that does not express ovalbumin antigen. So these are the controls. These mouse were immunized twice with ovalbumin containing liposomal formulation that has listrolysine O. When we challenge those animals with lethal dose of melanoma, then here is the data. OVA LLO containing animals, when they are challenged with MO4, meaning this is the murine melanoma B16 that expressing ovalbumin as an antigen, they are reasonably well protected, although the tumor size grows up as the days progress, meaning this is not a cure for the cancer. It essentially demonstrates the proof of the concept that if we can generate tumor antigen specific cytotoxic T lymphocyte response that slows the progression of tumor development significantly longer compared to all the control groups, they died within 2 weeks or so, but we did not see significant level of tumor development and progression until 3 to 4 weeks. Ultimately, these mouse also died. That data is translated into here. All the control group of animals died very early on, within 20 to 30 days, but some of the immunized and challenged mouse, they survived for a very long period of time. I tracked them up to, they did develop tumor. It's not that they did not develop the tumor. Here is the tumor progression, slowly developing about 3 weeks, and moving forward, their tumor size is growing. There's a square millimeter in diameter. Every alternate day, I will go into the mouse colony, measure with a calipers and record the data, and that data is plotted here. But some of those tumor-bearing mouse, I tracked them up to 100, 110 days. They still have a large tumor, but they are still surviving. Many of them died in early phases, of course. And at that point, I had to euthanize following a protocol, because you cannot really let them live with a large tumor mass on their back, because it is considered inhuman or cruel. So taken together, what these two data shows that if we generate tumor antigen specific cytotoxic T lymphocyte responses, we can slow down the progression of the tumor, and many of those slowed down tumor progressed individuals, maybe, or at least in the animal, they live longer. We were happy to repeat that information into LCMV, lymphocytic choriomeningitis virus infection model, where mouse was immunized twice with LCMV specific nucleoprotein, then lethally challenged with intracerebral injection of lethal dose of lymphocytic choriomeningitis virus. And here's the data. Elelo containing liposomal delivery of nucleoprotein of LCMV protects the animal at 100% level, whereas when there is no listrolysine O, but simply without liposomal NP containing vehicle containing immunization in the mouse, when they're challenged with lethal LCMV challenge, that protection goes up to 50% level. So that's about twice as much protective compared to the control group of animals. So taken together, what we could demonstrate that when we incorporate a bacterial toxin such as listrolysine O into the pH-sensitive liposome, we can trigger ovalbumin or nucleoprotein-specific cytotoxic T lymphocyte responses that protected completely against lethal lymphocytic choriomeningitis virus infection, or it successfully prevented melanoma cancer development, and many of the mice actually survived many months longer compared to the unimmunized mouse. So that's the take-home message. Some of the future directions we were working on trying to understand those are CD8 T cells, but are there any role of CD4s? The ratio of CD4s release cytokines, and those cytokines is actually energizes CD8 T cells, particularly interferon gamma, IL-2, those are, and interleukin 12. Those are the cytokines that are heavily used in cancer immunotherapy protocols these days. So those are some of the things we were interested in: development of vaccine adjuvants, particularly targeting innate immune receptors such as toll-like receptors, and subsequently, could we extend our observation into mucosal delivery, because many cancer developed in our mucosal layer. So this is the conclusion slide. What the data I have shown, been able to share with you in a little rapid fashion, is that Elelo containing liposome triggered cytotoxic T cell activity at a higher level with enhanced frequency. Those Elelo specific immunization containing ovalbumin or NP protected the mouse when they were challenged with murine melanoma cell types that express ovalbumin as a surrogate antigen, or lymphocytic choriomeningitis virus antigen NP, that also provided protection to those immunized mouse. We demonstrated that Elelo containing liposome, these are pH sensitive liposome, acts as an efficient vaccine carrier that are capable of priming anti-tumor, cancer antigen specific, antiviral protective immunity. And we have some data that shows that although we do generate listrolysine O specific immunity in those immunization protocol, yet Elelo specific pre-existing immunity does not abrogate vaccine antigen-specific immune responses. Because we have to remember that many of us gets foodborne Listeria infection. So there are chances that someone can have Listeria specific antibody or immune responses. That's all the data I had to share with you. I acknowledge Nehru Fulbright Scholarship Agency provided my scholarship so that I could come in here, and BITS Learning kind enough to host me with my fellowship. And these are my collaborators at UFM, Emory University, Atlanta, Rufi Ahmed and John Hueri. They are one of the stalwarts in CD8 T cell research in the context of viral infection and tumor antigen specific vaccines. And Dan Portnoy provided us at Berkeley, California, provided us with he's the pioneer of Listeria research. I had a Upjohn Valtech grant at that time, and also NIH funds provided to College of Pharmacy at that time. Thank you, and I will entertain any question if you have for me.
Thank you for that wonderful speech on your vaccine expertise. So we all learned quite a lot. Hope the participants have any questions. Not on vaccines.
Okay, that's all right. This is very specific niche area of research, but
So,
this
but
is
I have a
essentially
question.
yes.
Like,
Please go ahead.
I don't know about how you launch vaccines in US, but if I have to, if you're producing a vaccine and it has to be launched, what sort of procedure or protocol, or do you face any obstacle here? Like, is there any?
Yeah, sure.
Can you share some thought process?
Right. So first disclaimer, I never worked with industry.
Okay.
This is academic research.
Okay.
We actually develop the concept and we provide the proof of the concept, experiments, publications, research. If it can be scalable and if can be commercialized, then only they go to the commercial entity. And also, of course, many pharmaceutical companies, they are also pioneering cancer immunotherapy, CAR T cell therapy, and etc., cellular therapy for cancer. It's not exclusively for academia, or it's not exclusive for the industry. So the ideal is that commercialization of academic research into the commercial domain. So within the university setup in US, we have so-called incubators, idea incubators, and we have a technology transfer department where a promising technology can be transferred to intellectual property value there. When that is established, a partnership is required, or in US, we have different types of grant, SBIR grant, Small Business Development Grant. I'm sure in India with DST or Department of Biotechnology, that type of commercial development grants are available. So that will be the first step. Second thing one needs to do, I have shown you the data on mouse model, but human being is not mouse. An unfortunate side of cancer immunotherapy, cancer treatment is what works in cell line, Petri dish, that does not work most of the time in animal model. I was lucky it did work. And what works in mouse, that does not work in primate model, higher animal model. And what works in higher animal model, that does not work in usually human being. That's very standard all across any pharmaceutical research, particularly in the context of cancer therapy, cancer immunotherapy. It's a very, very difficult proposal. We can show many cell killing in a Petri dish. If you don't treat cells, they die. You don't have to provide them anything, treat them with anything. Forget about cytotoxic drug or any novel molecule. You just grow some cancerous cells. In 3 days, they will die on their own happily. Don't have to do any research. But unfortunately, majority of the cancer research starts there, ends there. To translate that cell-killing activity in Petri dish is tremendously difficult to demonstrate in a tumor model in mouse. More difficult in higher animal, extremely difficult in human, because then we are talking about clinical protocol, writing of IRB approval, clinical trial, clinical protocol, essentially recruiting patient population, with cancer, with all the inclusion exclusion criteria, and that's a tremendous effort. Very few academic institutions in the world are capable of doing that, but that's the shorter pathway, taking these approaches from academic research in the lab that we do usually, then slowly, step by step, going into the commercialization, and prior to that, clinical trials. If it works, it might take 10, 15, 20 years most of the time, like CTL specific immune checkpoint inhibitor drugs, Keytruda, Mevlimumab, and Ipilimumab or Yervoy. That's the first line of therapy. Jim Allison at MD Anderson Cancer Research Center in Houston working on that aspect for last 20 plus years, 28 years. So there are many dead bodies along the path. Very few times it will work. In academic setup, what I personally believe that we should, if we have a good idea, we should be able to demonstrate that idea beyond cell culture, beyond petri dish, into an animal model, and show the demonstration efficacy with a cutting edge analytical observation using various established technology, commercially available technology. So all those tetramer binding assay, ELISpot assay, CTL assay I was doing at that time, those are readily available commercially. Yeah. So that's where you begin. Then you narrow down your research interest, follow using your thought process, pinpointing to the keywords. Then comes the resources. You can only do experiments if you have the physical resources availability. So it should ideally begin with a lab that is interested, that has the physical resources available at the university level or at the collaborators level. Only then it makes sense to go for the search of the literature, going for cancer immunotherapy research. So it's a rather reverse process. First, you have to find a mentor or a senior scientist, faculty, who has an interest in cancer immunotherapy, CAR T cell, for example. That's what we were talking here today, who does this type of research. And you have to talk to that individual, that would you be willing to mentor me? Would you be willing to provide support me? Give me a spot in your lab. These are my thoughts. So while you are reading, you are enriching your knowledge base in a specific area of research, be it a vaccine research, be it a immunology or virology research or CAR T cell research. You have to do your homework first. Then you find someone who will support your interest. And at the same time, you're utilizing the library resources. Go to the database librarian at Bates, and you can actually do on your own exhaustive literature search. If you go into the NCBI, which is a freely available database search engine all over the world, all you have to do, register yourself at the max. Even without registration as a guest, you can do just put some keyword.
Okay, thank you, Dr. Manas Mandal, for your valuable lecture on Quest for T-cell vaccine. Now we have Professor Sundar Baral sir on behalf of Department of BITS Pilani, KK Birla Goa Campus Chemical Engineering. He'll be giving us the vote of thanks for participants and speakers who have attended this workshop. Baral sir, over to you.
Good evening to one and all present here, honorable speakers, professors of BITS, PhD scholars, and all the participants. It is an honor to get the opportunity to thank you all dignitaries. On behalf of the Chemical Engineering Department, BITS Pilani KK Birla Goa campus, I would like to express my gratitude to all esteemed delegates of the workshop for their presence and contribution to making this online workshop a great success. I extend my gratitude to our honorable speakers for taking out time from busy schedule to grace the event with their knowledge and experience. Thank you for your inspiring and encouraging us with your words on this special day. I extend my gratitude to Dr. Jagata Nambikrishnan, Assistant Professor of the Chemical Engineering Department, for organizing such an informative webinar. I must thank the organizing team both Mr. Sudhir and Mr. Sarthar, and volunteers for working hard for the past few weeks to make this workshop success. I am thankful to the participants for their active participation. Thank you everyone once again for making it a great success.